What we're going to talk to you a little bit about today is what we're trying to do at FAST, and then about our FIRE initiative. And we have 3 of the 4 inaugural scientists with us today from the FIRE initiative, and each of them is going to get up and tell you a little bit about themselves and how they got involved in Angelman Syndrome research, and then about what they're doing with this initiative. I just wanted to give a brief introduction to what we're trying to do. My name is Becky Burdine. I am your Chief Science Officer for this foundation, so I work for all of you. And my job at the foundation is to get the grant proposals that come in and make sure that they look like something FAST wants to fund. And then I send them out to our scientific advisory board, and these 3 men have been on our scientific advisory board, or will be hopefully on our scientific advisory board soon, along with 10 other very distinguished individuals. And they've all been very gracious about reading our grants, going through them, making sure they're good. They've been incredibly rigorous, so you should be very confident that the science we end up funding is science that many scientists have looked at and have said, yes, this is where we think you should spend your money. Because I think, as all of you know, we feel like every dollar we have at FAST is precious, and it represents something that could be important for our kids, and we want to make sure we spend it in the right place. So I said this last night, and it's true. In 2008, Paula decided that she really wanted to form this foundation, and she wanted to make sure the science was going at the fastest rate it could possibly go so that our kids could have every opportunity to live the lives they wanted to lead. And I put this up here because I think when she first did this, I think a lot of people were saying, you can't do that, and you're not going to be able to do that. And I think it's because people are worried that you are going to do it. And I'm still really thrilled that that letter that Paula wrote in 2008, all the things she outlined in that letter we've done. And that includes, and she said in that letter, I want to have a clinical trial for a treatment in Angelman Syndrome. And I read the letter in 2008 and thought, I want that too, but I don't think I believed as much as Paula did that we can make that happen. And when I say we, I mean everyone in this room, because every one of us voted in the contest, or we've supported each other, we've done all kinds of things. This is the community, and we are your foundation. Okay, so why do we think that there is a therapeutic out there, and why do we think we can cure this potentially? And you know, I don't like cure, and we have lots of arguments on Facebook about what cure means, so let's do it this way. How do we find a meaningful therapeutic for Angelman Syndrome? That's what we'll say. All right. So most of you know the history, but I'll just review it in case you're new in the room or haven't thought about it in a while. In 1965, Harry Angelman described 3 cases, and he called them puppet children, which of course we don't like. But he had 3 children in his practice that all seemed very similar. Now, I think most of you in the room probably recognize this was an interesting coincidence. Some of you have probably never met another child with Angelman syndrome. Some of you probably live in the vicinity of 3 or 4. Right? It's a coincidence how many of them end up in the same area, but he ended up with 3 kids, not dissimilar in age, that all had similar characteristics, and that's why he could describe this as a syndrome. It took almost 20 more years before Ellen McGinnis identified the fact that children with Angelman syndrome had deletions on chromosome 15. 10 years after that, it became clear that the gene on chromosome 15 that was important in Angelman syndrome was the gene UBE3A, which we've talked about before. This is a ubiquitin ligase. Its job is to go around a cell and find proteins that should be degraded and put a moiety on them called ubiquitin. So it basically takes this little protein called ubiquitin, sticks it onto proteins, and that tells the cell that protein's life is over, get rid of it, recycle it, and move on. It has other functions too. Scott may tell you about some of them, but that's the main thought of what we think UBE3A does. And so when you don't have UBE3A, the thought is that your cells are now filled up with proteins that shouldn't be there, and essentially your cells are trying to work in the presence of lots and lots of garbage. But this is one of the reasons why we think that there's a potential meaningful therapeutic out there. We now know the gene that causes Angelman syndrome. And most of you in the room have been hearing about this gene for a long time, so you may not understand the significance of that. But there are lots of syndromes out there, and you can think of autism as being one of them, where we don't know what's causing these things. We don't know. And so you can say, my child has autism, but I don't know why. And if you don't know why, there's nothing you can think about really meaningful to try to do to treat it. We know why our kids have Angelman syndrome, and we know what they're missing. And they're missing UBE3A. And so you'll see how fast things started to go after this. So we found the gene. Within a year, Yongwei Zhang had created this mouse model of Angelman syndrome that most of them use in their research. In 2007, Ed Weber and Ipa Elgersma, along with a lot of other really great scientists, showed that if you combined a mutation in UBE3A, so the mouse has Angelman syndrome, with a mutation in another gene, you could actually make those mice look like they didn't have Angelman syndrome anymore. This was mind-blowing, and this was really one of the impetuses that started the foundation, because we read this paper and we thought, wow, if they can actually make a mouse that should look like it has Angelman syndrome now look like it doesn't have it, why can't we do that for our children? Why can't we help our children? And that's one of the reasons why we founded the foundation, because we felt like with this finding that was reported in 2007, things should be moving faster. I still don't understand why the world isn't jumping up and down and saying, here's a neurological disorder that we might cure, and if we figure out how to do it, maybe we'll figure out good treatments for things like Alzheimer's or autism or other neurological disorders. I'm still amazed when people say, oh, I've never heard of Angelman syndrome. And I'm like, why have you not heard of this? This is a scientific breakthrough on, I can't even tell you how monumentous this is going to be when we finally do find these meaningful therapeutics, not only for us, but for the neurological research field as a whole. All right, so I'm just going to review really quickly because they're going to tell you about what they're going to do next. There are a number of known mechanisms that cause Angelman syndrome. So all of you are aware that typically you get a chromosome from your mom and a chromosome from your dad, so you have 2 copies of your chromosomes. This is a great backup mechanism for us because if you get a bad copy from, let's say, dad, I'm going to make the dads bad guys here. If you get a bad copy from your dad, you probably got a good copy from your mom, and so you don't know. If I sequenced all of your DNA in this room, you would have mutations all over your DNA, and they're not meaningful because you probably have a backup copy somewhere to compensate for that. The problem with Angelman syndrome is something called imprinting. Now, I am not blowing smoke at you when I say this. There are a lot of scientists out there that if you say imprinting, they go, ugh, and they don't even want to think about it. You guys think more about imprinting than a lot of people in the world. And that includes your doctors, and you know this because you've talked to them and you've explained it to them. What happens with imprinting, and we don't exactly know why things get imprinted or not imprinted, that's still something we're trying to work out, is that it turns off one of those copies. So instead of getting 2 good copies of something, you get 1 good copy. And in the case of Angelman syndrome, this region gets turned off if it comes from dad. So the chromosome that you get from dad, the region where UBE3A is, gets turned off, and the cells just don't recognize it. It's almost like it's not there. And it usually doesn't matter because you get this gene from your mom, and it's perfectly fine and functional as long as you get it from your mom. The problem with imprinting is if you don't get it from your mom, and for most of our loved ones it's a deletion, if you get a deletion of this region, so this gene's not there from your mom's chromosome, the cell still sees this as your dad's chromosome and it still keeps it off. So now you have no UBE3A, and this is why you get Angelman syndrome. There are other ways to do this, as you know. You can have a mutation in the mom's UBE3A, usually inherited from their fathers, and now there's no UBE3A, Angelman syndrome. In rare cases, mom's chromosome can be lost from the egg, and then you duplicate dad's so that you can survive, but now you have 2 copies that are recognized as not being functional and they're turned off. This is called uniparental disomy, or UPD. And then there are imprinting defects, where both chromosomes are fine, but the imprinting mechanism goes awry, and so mom's is not recognized as something that should be turned on. The reason I'm walking you all through, and I'm going to point out too that I went through that very fast. If you're new to this, or if you haven't thought about it in a while and you want to think more about it, if you go to this site on our webpage, if you go to the Cure Angelman webpage and go to the What is AS section, we've written a genetics 101 and a testing 101. This was written by myself and Aaron Sheldon and reviewed by Wen-Han Tan, that walks you through how you get these, what these different mutations are and what they mean, and how those testing results that you got from your kids give you which of these Angelman syndromes you have. And I don't want to forget the fact that we also have a class of kids in our community, loved ones in our community, where none of these apply. We do not know why they have all the characteristics of Angelman syndrome. These are our clinical cases. And we're going to talk a little bit more about those today, too. The reason I'm telling you all this is that the mechanism for Angelman syndrome is one of the reasons we think there's a therapeutic out there. Because recognize that in all the cells in your loved one's body, there's a perfectly good copy of UBE3A sitting there. It's just not on, but it's there, right? So imagine that Angelman syndrome was something where it was completely gone. It wasn't even in the genome. It was deletion on both chromosomes, let's say. We would have to go in and put it back. We would have to do something like stem cell therapy. We'd have to repair genomes. We would have to do stuff that it's in our capability, but we're not really doing that now. We're not doing that in the terms of clinical trials, really. In this case, we don't have to do anything. The DNA is sitting there. It has the gene. It's just not on. All we have to figure out how to do is turn it on. That's one of the ways we can cure AS. And I should say cure, meaningful therapeutic for AS. One of the other things we could potentially do is just bypass the fact that this gene is missing, figure out what it does, and compensate for that. So in 2011, we funded the study that Ed used to find the minocycline that showed that it could alleviate symptoms in the AS mouse. This, along with the fact that, again, from Ed's lab, they did viral replacement of UBE3A. So they used a modified cold virus, they put it into the mouse brain, and that put a UBE3A gene back in the brain that would be expressed, and those mice recovered from some of their symptoms. These two things tell us that, well, they tell us a lot of things, but one of the things I think is important here is these are adult mice. They put the virus into their brains, they recovered from some of their symptoms. I think what this tells us is there may not be a window, right? I was worried very early on with Sophie that maybe there was only a few years where I could do something, and then after that, I couldn't do anything. These are adult mice, and they recovered from some of their symptoms. I think that gives, at least me, some optimistic hope that if we find something that's meaningful, it will help children and loved ones no matter what their age. Ben Philpott's lab showed that using a cancer drug, topotecan, they could turn on UBE3A in the mouse. So we know from a proof of principle standpoint that you can do this. In 2012, the human clinical trial for minocycline began. We heard a little bit about that last night. We'll all just cross our fingers for the next few months until we can hear the results and hope that we're going in the right direction with that. And so what we're really going to talk to you about today is the FIRE initiative. This is the FAST Integrative Research Environment. I'm going to let Ed talk to you a little bit more about how this is going to work, but I'll leave this graphic up here while he's talking, so you can look at it if you want. But what we're doing is we are funding 4 inaugural researchers to come together, to work together, to synergize, and help us proceed even faster than we have before to try to find meaningful therapeutics for Angelman syndrome. As you know, Dr. Dindot, Dr. Siegel, and Dr. Weber are here to talk to you. Unfortunately, Dr. Anderson is at the huge epilepsy conference that happens every year, which we didn't know happens to fall right in the middle of the FAST Gala every year. Our bad. Okay, but, so she's at the epilepsy conference and couldn't be here, but Ed will tell you a little bit about what Dr. Anderson will be doing in terms of the FIRE initiative and how that's going to help us. So, and I should also mention that FAST is funding this. We've got the first year's funding set up. We're looking for more funding. And FAST Australia has decided that what they would like to do to contribute to the FIRE initiative is supply money for a postdoctoral fellowship. So what they're going to do is they're going to find an interested scientist in Australia that would like to come to the US and will do work on Angelman syndrome in the context of this initiative in one of these labs. And so they're going to provide the money for that and partner with the lab here in the US, and the hope is that that postdoc will come up with their own ideas and programs and go back to Australia and set up Angelman syndrome research there, because they would like to have more people doing Angelman syndrome research. If there are other organizations out there listening or in the room, and you would be interested in doing something like this, please drop me an email, give me a call. We would love to have other people involved in this. And like I said, I'm coming back to this. So how can you help us? Keep raising awareness. Of course, I'm showing you Sophie because I have permission to show her picture. This is my daughter. This is how I got into this. This is why I'm standing up here babbling on. My daughter Sophie is now 7. When she was 11 months old, she was diagnosed with Angelman syndrome. And very early on, within a day or two, I had contacted a friend of mine who worked on imprinting, because that was the only person I knew that had anything to do with Angelman syndrome. And she said, I'm going to have someone named Art Bodette call you tonight at home. And I said, okay, fine. And I waited for his phone call. And he called me and he said, what do you want to know? And I said, well, I want to know everything. What's going on? What's going on with research? I'm a developmental biologist, and so I've been trained with the idea that if something goes wrong in development and you don't develop correctly, that's it. Right? I see this happen all the time. I work on congenital heart defects, and people are always like, are you going to be able to cure them? And I'm like, no, I'll be able to tell you why it happened, but I'm not going to be able to fix it. A surgeon can fix it, but I can't. So I was ready for some really tough news. I had already prepared myself. And the first thing out of Art's mouth was, are you in the new clinical trial for betaine folate? Because the research in Angelman syndrome is amazing, and there are potential therapeutics down the road. And I'm on the phone going, what? And I got off the phone with him after about an hour and a half. He was at a conference and talked to me for an hour and a half. I couldn't believe it. And I went into my husband, and I'm like, I don't understand the phone call that I just had. And I tried to tell him, and he looked at me like, are you drinking? Because he's a scientist too. And he said, I don't think you heard him correctly. And I'm like, no, I did. I heard him correctly. And the next week I called Joe Wagstaff and I said, Joe, you got to put me to work because I don't understand what's going on. And so I got in, started reading the science, and realized that everything Art Bodette told me was true. There was a tremendous amount of hope out there, and the scientists thought there was hope too. But what struck me when I got into the community is that most of the people that I met in the community had no idea that any of this was true. No one knew that there was this excitement. Nobody knew about the science. Nobody knew that Art Bodette was telling me that there were therapeutics. And I felt like it was very unfair that I had that access, that I had the advantage of being able to call these people because they were scientists and we were scientists and they trusted me as a scientist. I didn't think that was fair. I felt like everybody should have access to that information, especially because it's our loved ones, right? I don't think everyone should be fighting that hard to figure out what it is they need to know. And so we've been very committed at F.A.S.T. I hope we've done a good job of this, of trying to make sure that you get the information that we have available to give you. We don't hold anything back from you. If we have it and we can give it to you, we give it to you. Sometimes you may not want to hear it, but we do. And so if you have questions, if there are things you want more answers to, let me know. I can try. But I don't want anyone in this room to feel like they don't have access to stuff that they need to live a life that's better for them and better for their loved ones and their children. All right, so the last thing you can do after all that is we need money. It keeps me awake at night sometimes to realize the thing that stands between my daughter and a life with no seizures is money. You know, I watch the Super Bowl commercials every year and I think, that's the difference between my kid and a life without epilepsy. That's the difference between my kid and the ability to walk across a room and get a drink. It drives me insane. But we're a rare disease. Nobody's going to make any money off of us. I'm sorry, they're just not. And so it's hard to say to somebody, this is what you need to work on, because they have a bottom line, they have a business, they have people they answer to, they have employees that have to keep working. Nobody's being cruel about this. This is just how it works. So we need you to help us. We're trying to identify people who might be willing to get behind us with this, behind this initiative, help us find these therapeutics. I'm willing to go anywhere and talk to anybody. I'm willing to take them out for drinks, whatever we have to do to convince them that we're serious about this, that we know what we're doing, and that they have the potential to fund a cure in a neurological disorder that may not just help our kids, it may help them someday, because I did this with the high school students at Eastside. I'm like, how many of you have ever heard of Angelman Syndrome before you started doing this fundraising? None of them. How many of you have ever heard of Alzheimer's? Ching. How many of your parents or grandparents are worried about Alzheimer's? Everybody's worried about Alzheimer's. This Angelman syndrome is a defect in learning and memory, and that is what Alzheimer's is. The mechanisms are completely different, but imagine if we found therapeutics that could increase the ability of our kids to learn. How is that not potentially going to help us figure out how to help people with Alzheimer's not lose their ability to learn? So we're looking for people who want a legacy. We're looking for people who would like to know that their money went to something that was going to work. Now, you hear this all the time, right? Give us money for cancer because we're going to cure it. Give us money for heart disease, we're going to cure it. Give us money for leukemia, lupus, we're going to cure it. And they've had a lot of money and you've seen no cures, and that's because it's hard. It really is hard. We've got science on our side where they don't, in some cases. We're not talking about a lot of money. I think if we had $10 million, we could run FIRE for 5 years, and I can't imagine what could come out of that, but I think it would be fantastic. You know, with a very small amount of money, we found minocycline and did the clinical trial. Imagine what we could do if we actually had adequate funds to give these 3 gentlemen and Ann Anderson the ability to hire enough people to do the work that needs to be done. So we don't do it in piecemeal pieces. We do it right now with all of the manpower that we need, or woman power. Okay, so that being said, if you have any great ideas, we're not asking you to get your money out. Nobody needs to go bankrupt in this room for this, and I mean that. If someone tells you this is the therapy that's going to make your kid better, call me and I'll tell you if I think it's a snake oil salesman or not. I'm very honest about that. Nobody should be spending their own money if you can't. What we're looking for are people who have the money to spend and might be willing to do so. So if you know somebody, if you've got an idea, if you want to run a bake sale, whatever you want to do, we're happy to let you be part of this and to help us. All right. I'll go back to this, and now I'm going to turn it over to Ed Weber. Michael's going to move the live feed so that the people at home can see Ed and Scott and David. While they're doing that, if anybody has any questions for me, I'll answer them very quickly. We are filming this, so if you're going to ask a question, we ask you to go to the mic so that people at home can hear what your questions are. We're only going to do it for a few minutes because I want you to hear the really important people talk today. And after they've talked, we're going to open it up for everybody. You can ask questions of anybody. But while Michael's moving this, we'll go ahead and do a few questions if you've got any. And it's okay if you don't. Okay. So, hi.
Hello. And if you want to do fundraising beyond bake sales and such, is there a legal procedure that one has to follow, like register as a fundraiser just to do it legally?
Is Danielle in the room? Where's our event person? She was in the room. Danielle, do you have to register to do anything legal? Can you come to the microphone if you're going to? This is Danielle Penders that's coming up to the microphone. She's our newest board member, and she is in charge of events. Why don't you tell them a little bit about you before you answer his question?
Sure. Well, I've been in development for several years for several national nonprofits in fundraising and development and marketing, and so I have a pretty good background in understanding about how to run events and how to do third-party events and attract corporate sponsors, things like that. So if you're certainly interested and have ideas or have people that you feel would be potential funders, I'm more than happy to be accessible to talk about those things, walk you through how to get an event up and running for F.A.S.T. You don't need to be you don't have to be registered to do an event. You just there's some materials that we would work with you on just to get things in line, but feel free to contact me at any time, and I can help you get going.
And that your email is events@cureangelman.org.
Yes.
Correct?
Yep.
So Danielle can help you figure out if there's anything you need to do that would be along those lines. Thank you. Karen. See if it's pushed up to on.
Betaine folate study. And I'm just wondering, what was the mechanism or the theory behind that, if you could explain it to me?
So in a nutshell, the idea is that imprinting is controlled by something called methylation. So it's a modification of DNA that determines whether or not the DNA is going to be turned on or turned off in terms of making a gene product. The betaine folate trial, the idea was that if we put enough B vitamins into somebody's system, maybe they would make more methylation events happen, and that would change the imprinting at the locus. It wasn't a bad idea, but we didn't get a good result from that trial. We didn't get any result from that trial was the problem. So that didn't seem like it was the way to go. But the general idea there was to try to reverse the imprinting and make the paternal come on, just like what we're trying to do. So that was the hope, is what that would do.
I do have one question. With the research, is the research to eliminate just the epilepsy or to reverse the whole syndrome itself where, our children can maybe walk a little better, talk, or is it just to eliminate the seizures?
As a parent, what I would like is for all of the characteristics that prevent my daughter from living the life that she would like to lead to be gone. And for my daughter, that would be the ability to walk, the ability to use her fine motor skills, the loss of epilepsy. Those are the three big ones if I think about it. It would be good if she could talk, but we can get around that, right? I can get around the fact that she doesn't talk. It's hard for me to get around the fact that I could lose her to seizures. And so when people, approach me and they say things like, I don't want therapeutics, I don't know why you're wasting your time with therapeutics, I get it, I really do get it, but I've almost lost my daughter twice. And there are people who are not so fortunate that are not sitting in this room who have lost their children. And I don't want to I'm not scaring anybody for that. I'm just saying that epilepsy is, I think everyone in this room who's dealt with seizures will tell you that it's the worst thing. I could deal with almost anything if she didn't have seizures. The seizures I cannot deal with. And that's just me personally. So when I said that, I was talking about my own experience. I want her not to have epilepsy. I really don't want her to have that. And so if we found a therapeutic that just eliminated that, I could probably live with the rest of it and I could make her a phenomenal life. But I would like to see it all gone. In a perfect world, I'd love her to pop a pill and no longer have Angelman syndrome. And I know there are people out there that don't like that idea, but I would be lying to you if I said that wasn't the truth. Okay, yep. All right, I'm going to turn it over to Ed. You guys want to, would the three of you like to just introduce yourselves and then you can talk? Okay.
Oh, go ahead. You can start. Since you're on
Are we all gonna?
Yeah, go ahead and then.
Okay, I guess I'll start. My name is Dave Siegel from UC Davis out in California. It's an honor and humbling to be able to work for you now. I'll just make a quick introduction before I talk more about what we're planning to do in the FIRE grant. But I started out in New York, grew up in New York, went to Cornell University for an undergraduate, got a PhD. Ah, see, there's some people. Got a PhD at the University of Utah in Salt Lake City, and then did a postdoc, which is after the doctorate, out in San Diego, California, at the Scripps Research Institute. And I'm now an associate professor at the University of California at Davis in the Department of Biochemistry and Molecular Medicine. And I think that's, I'll just... How did you get into it? Okay. Actually, that's a very interesting story. So I think it was about 5 years ago when I was contacted, and I checked my fax with Paula Evans, and she reminded me, it was her that contacted me. Yeah, more or less. So I was just minding my business, working on my science, and I was contacted really out of the blue by Paula Evans and some friends, concerned groups of parents and researchers from a Google group, and they were interested in the kind of work that I do, which is engineering DNA binding proteins as potential tools for research and therapeutics, which I'll talk a little bit more about, I guess, soon. And they thought that maybe this technology could be used for Angelman Syndrome research. And at that time, I have to admit, I didn't know anything about Angelman research or Angelman Syndrome, and I had to look it up on that vast repository of all human knowledge, Wikipedia. And so I looked it up, and I saw what I thought I understood, and I immediately wrote back to them, and I said, look, I'm very sorry, but this is there's a protein that's missing, and it's probably missing throughout development, and so I don't know if there's a late-stage intervention that we could do that would make any difference. And right away, they sent me this paper by this guy named Dr. Weber. And the paper said that it's not a developmental defect like that it's really a signaling defect. And if we could get that signaling going again, we could really, even at a late stage, have a real therapeutic benefit. And that's what really got me started in all of this. And it's really changed the focus of my research. Thanks.
That's going to be hard to So my name is Scott Dindo. As Dave said, it's a pleasure to be here. I'm honored to be here, and it's been a very humbling experience to be here. So thank you all for inviting me. I think all these guys feel the same way as I do. So currently I'm an assistant professor at Texas A&M University. I have a lab that most of the research that we do involves Angelman Syndrome. And so there are a number of other diseases that we study, but Angelman Syndrome is the primary disease that we study. My exposure and involvement in beginnings in Angelman Syndrome began when I started my postdoctoral fellowship. So I have a PhD in genetics from Texas A&M and went on to Baylor College of Medicine in Dr. Art Baudet's lab, where I spent 5 or 6 years training there. And so while I was there, we worked on trying to understand the mechanisms that regulate the expression of UBE3A, and they were also interested in understanding what the function of UBE3A is in the brain. And so as has been mentioned here, it's a fairly complex gene in terms of the way it's expressed. And so we've been trying to dissect that process, or I have for the better part of the last decade. And so my involvement in this comes from our knowledge of that gene and how it's regulated and what it does in the brain and so forth. But again, it's a pleasure to be here, and hopefully we'll have time to talk about the stuff that we're doing quickly.
Don't worry, these guys will get used to this as I have. For those of you that I don't know, and the first time people here, my name is Ed Weber. I'm a professor in the molecular Department of Molecular Pharmacology and Physiology at the University of South Florida. I'm also the Chief Scientific Officer at the BIRD Research Institute, Alzheimer's Research Institute, where my laboratory is at the University of South Florida. A lot of people ask why I got involved and how I got involved. It was kind of weird circumstance that I was a postdoc at Baylor College of Medicine a little bit before Scott was there. And my laboratory, or at least the laboratory that I was working in with J. David Sweat, who was my mentor at the time, was right next to Art Baudet's lab, who was looking at Angelman syndrome, and along with Joe Wagstaff and some other researchers were the first to identify the UBE3A gene as being the AS gene. So they found the gene, they had developed the mouse model, and for me as a burgeoning neuroscientist and understanding what was happening in the brain and in learning and memory, this was really an exciting thing. So before that, how you did neuroscience is you went about just knocking out genes and seeing what happened. And this was the first time where they were actually trying to mimic a human disorder in a model that they could do research on. So it was really an innovative research. Now, when I left Baylor College of Medicine and went to Vanderbilt, David Sweat and Art Baudet were gracious enough to allow me to bring the mouse with me. And I always get this question, was it just one mouse? So when we say the mouse, we mean the mouse model. So we have hundreds of these mice that we do a lot of different research on. So it wasn't just one single mouse that I brought with me. It was actually two, a male and a female. And from them, you know. So when I went to Vanderbilt, the interesting thing is that I had this crazy idea of doing this double mutation to try to rescue the mouse. And a lot of people thought I was crazy. I thought I was crazy. And I had graduate students that were coming in that were doing other work, and I would say, we could do this, you could breed these mice, you could, it'll take a couple years, but it'll be, and they're like, oh God, we don't want to do this. So it was actually an undergraduate that came in that didn't know any better, and she was like, yeah, sure, whatever, I'll go ahead and do this. And when we bred the mouse together, the CAMK2 mutant and the AS mouse, we really knew we were onto something, and that's what caused a lot of this buzz that's around. After I did that and published that paper, I was quite convinced that I would finally be through with Angelman syndrome. I'd finally got somebody to do the experiment, and I could concentrate on what I was doing at the time, which is lipoprotein receptor research and how it is associated with Alzheimer's disease. And like Dave Siegel, I was contacted by Paula Evans and a number of other different parents that started asking the questions of what would be next, what would I be doing next, what did this mean to the greater field of science, and how could this be developed into a therapeutic? And those were all questions that I had never thought of before. I mean, as a scientific researcher, you don't think along those lines. And so the Paula and the parents and Becky, they really got me to start thinking differently about my research and how I did things. So this is how I got to be here, and very supportive of F.A.S.T. And why I'm here in front of you. So before we go on to the fire, unfortunately Anne couldn't be here. She would be sitting right here. She was actually chairing several of the sessions in this epilepsy conference, which is very big, which if nothing else exemplifies where she's at in the field. She is an expert in pediatric neurology. She's an expert in epilepsy. She's the director of the seizure monitoring unit at Texas Children's Hospital. She's also an associate professor at Baylor College of Medicine, where it's funny how we're all kind of tied together. I've actually known Anne for a number of years, and she worked in Dave Sweat's lab when I was a postdoc at the time. She's a clinician. She's a medical doctor who also does science as well. So she has a very large patient base, yet she also has a research program. So she is very interested in how seizures are produced. And one of the things that we don't know is how the absence of UBE3A causes seizures in Angelman syndrome. I mean, it seems crazy that we don't know this, but it's still really a big black box there. And so when we decided to start thinking about generating a consortia of researchers, And you can probably see after talking with these guys, it was pretty clear that these were the people that we felt would be great to be working in a team, sharing data directly. And as you can see with the kind of graphic that we have up here, when you think about how science is done, usually how science is done is, Scott here, he's a geneticist, he's working on genes, he's looking at Angelman Syndrome. He does work for a year, it takes him 6 months to get his paper published. I finally get the paper across my desk and it'll pop up and maybe 3 months later. So the research and the experiment that he did to make this really cool finding, I'm not seeing for another 1.5 years. Okay, so I look at this and I go, oh, this is pretty cool. We should integrate this into our research. So it takes 3 or 4 months to get that integrated. Then we do the experiment. I find somebody stupid enough to do the experiment. And then, we publish the paper, and then 1.5 years later, Dave gets that paper and goes, oh, this is pretty cool. And he worked off. So you can see how this can really stretch out how scientific discovery is made. Now, we collaborate, and there's a lot of researchers that collaborate. And then I would say that the AS field, the Angelman Syndrome researchers that are out there, which is a fairly small group, are probably more interactive and more collaborative than any other area of research that I'm involved in. So we do collaborate and we do share information, but it's not shared in real time. So if we had a situation where multiple investigators can get together, can share information in real time to where he does the experiment and he calls me up and says, this is something cool that you should be looking at. You can see where that can shorten the time into scientific discovery, and it can also shorten the time where we can identify, evaluate, and get therapeutics into the kids as quickly as possible. So this was kind of the brainchild for the FIRE grant and this kind of initiative. And as you can see, we've got, we have a geneticist. I'm involved in learning and memory. I do a lot of behavioral research. We've got Dave Siegel over here who's doing some incredibly cutting edge research with the artificial transcription factors. And I have to say another thing too is that his research and the techniques that he's using could be much more applicable to other disorders. And yet he's decided to do this with Angelman syndrome. So, I feel privileged and blessed in a lot of different areas in my life, and knowing a lot of you that are out there in the audience right now is one of those. And I feel very privileged to be able to stand here and talk about this initiative and be able to work with these guys on really almost a daily basis now. So we're excited about where we can go, and I think that what we'll do now is just kind of talk about what each of us is thinking about doing in the FIRE grant, and we'll send it back to Dave Siegel, and he can tell you what he's thinking about with artificial transcription factors.
Okay.
Thank you.
Okay. Well, that was warm and generous. So I just want to make one point before I begin. That is, the story about how I got involved and Ed got involved and I guess Scott got involved to some extent. It really has to do with all of you, with the parents. And I know that, a lot of times, you might wonder what role you could play in the scientific process, and it often comes down to giving out money. But I just want to tell you, it's not just that. So, when you talk to scientists, when you contact them, when you ask them questions, when you show an interest in their work, tell them about yourself, about your story, it matters. It matters a lot. So you should know that. Yeah, thank you. So I'd like to tell you a little bit about some of the interesting things that we're planning for this FIRE initiative. I think it's very exciting. So the technology that I work on that Dr. Weaver was referring to is a method to try to develop proteins that can turn genes on and turn genes off. Now, in nature, in our cells, we have proteins that do this for a living. They're called transcription factors. And the transcription factors have some way of recognizing what gene they're going to bind to and turn that on or turn it off. And so over time, we, meaning me and my other community of people, certainly not just my lab, have studied these transcription factors. We've studied carefully how they bind to the DNA, how they recognize their targets. And then over maybe 2 decades, we figured out how we could reprogram them to bind to sequences that we're interested in. For example, sequences involved in Angelman Syndrome. And in that way, we think we have a capability to go in and actually turn on a gene that we're interested in or turn one off. Now, this has some very exciting potential in a lot of different areas. And I just want to make a comparison to put this in perspective. So when you have a drug, drugs usually target proteins, and proteins come from genes. So genes are making RNA, and then they make protein. And you know, the strongest weapon that we have in our arsenal today is drugs, for many reasons. They get into cells well. We know a lot about them. We can't always make a good drug for every protein target, but that's something, you know, we have a lot of drugs for a lot of protein targets. We can develop more. People are familiar with drugs. Doctors are familiar with drugs. We know that there's side effects. We know how to look for things like that. So, you know, the drug technology is very established and for good reason. What I'm describing is a little bit different. We're not going to target drugs. We're targeting the gene. And since Angelman syndrome is a genetic disorder, ultimately, the ability to go in and target particular genes that are involved, for example, UBE3A, should allow us to make potential therapeutics that are very specific. So that part is very exciting. Now, what are the challenges there? Well, it's not a small molecule drug. It's a protein. We need to get it into the body. We need to get it into the brain. And then we need to get it into the brain cells. So there are some proteins that are used as drugs today. Most of them are antibodies. But there are also some of these artificial transcription factors in clinical trial for other disorders, not from my lab. So I think, you know, there is a real opportunity here to try to apply this for Angelman syndrome to reactivate the silenced UBE3A gene on the paternal chromosome. So that's kind of what I'm trying to do in a nutshell. And recently we've had some promising results in a mouse model where we're trying to do the same thing. That's not been published yet, but we're still working on that. And I think that's given us a lot of hope in our lab that this is something that we could really develop. The only other things that I would mention about the F.A.S.T project is that in addition to trying to develop drug therapeutics, protein therapeutics, whatever we can develop, it's important that we have a good way to test these potential therapeutics. We don't want to just go out and test them in people, especially when it's very experimental. So we need animal models for that. Before starting on this Angelman syndrome work, I used to pride myself in saying, I've never worked with anything larger than a cell. And you know, realizing that, you know, I really have to translate this to the next level has really moved me into this area of mouse research where these guys have been working for a long time. And so we've been using an Angelman syndrome mouse model. And that's been very helpful, and I think it's been very helpful for all of us in this field. But, you know, thinking back, looking historically, why do we use mice? We use mice primarily because some Nobel Prize winning work has allowed us to efficiently make genetic modifications in mice. This has been a tremendous boost across the biomedical spectrum of research to be able to use mouse models of human disease and to be able to, you know, create these genetic models of disease. But new tools are emerging, and mice are not the only show in town anymore. And in fact, there's some animal models that are better than mice. Rat, for example. It doesn't seem like it's that different than a mouse. It kind of looks like a big mouse. But in many ways, rats are a better model for neurologic disease than mice are. Their cognitive abilities are somewhat, you know, more attuned to what we're really trying to do. And so one of the things that we're going to try to do is apply this newer technology to make the very first Angelman syndrome model in a rat with a large deletion. So that's one goal. And another goal, aside from better animal, and you know, when I talk to my colleagues at Davis, you know, Davis is an ag campus, and they think big. So they say, you know, you don't have to stop at rats. There's pigs. Pigs are even closer. Now we're not looking to make a a model pig at this point. Pigs are a little hard to work with. They might be a better model, but they're kind of big. It's hard to put a pig in a Morris water maze and measure how it swims. So, but I just want to say there's other kinds of animal models, and we're going to apply this newest technology for Angelman syndrome research now in the fire grant. And then apart from animal models, we'd like to work in things that are closer to human without working in actual humans at first. So the closest we can do that with is cells. And we're also at a very exciting time, when we can use induced pluripotent stem cells, which is another cutting-edge technology, and use those to actually turn them into neurons in a culture dish and show that we can make changes to those cells in a culture dish. Now, culture dish is not a brain, so we still need our animal models, but these new human cell technologies will allow us to test our potential therapeutics in human cells too. So I'm going to be involved in that part as well. Thanks.
Can you put up that slide with the chromosomes? So while she's working on that, I'll talk briefly about sort of our role in this project and what strengths and expertise we bring and sort of the questions that we're trying to ask. Yeah, if you can put them all up. So can the slide be turned on, the projector? So my lab is interested in my training and so forth has been to try to understand how this gene is regulated in the brain. And so there's like I mentioned, I sort of argue that it's probably one of the most complex genes in the human genome because of what happens. But because of that, there's this opportunity for this therapy that Becky's talked about. And so as she's mentioned, all of us in this room, the allele that we inherited from our mother is the one that's being expressed, and the one that we inherited from our father has been turned off. And so it's not entirely clear why that happened. I mean, there's about 100 million years of evolution that drove that to happen, but nonetheless it does. But it provides an opportunity to develop a therapeutic because there is a normal copy that's there. And so we just got to find a way to awaken that allele that's been turned off. And so one part of this project that we're going to be involved in is just trying to understand this mechanism. So how is this gene turned off, or how is this allele turned off, and why is it turned off? And if we understand that, then that will open doors for us to develop potential new therapeutics. We really don't know anything about this gene. I mean, we know some things, but if you compare it to, let's say, other genes that cause diseases and so forth, we're just not there yet. And so a large part of what we're going to be doing is trying to understand how that paternal allele is silenced in neurons. And so we've developed some unique tools in mice, and we have some very unique tools at Texas A&M where we can try to dissect this process to figure out how this gene is regulated. So I think if we can do this, it'll open up lots of possibilities in terms of maybe just not reactivating this gene, but just understanding what it does in the brain. Because although we kind of know, the general purpose of this protein, we really don't know what it's doing. I mean, there's a lot of debate right now in the field what exactly it does in the brain and why it leads to the symptoms it does when it's gone. So the second component that we're going to be working on is it's directly related to the first, and that is we're just going to take an unbiased approach to look at drugs that may reactivate that paternal allele. So this has already been done by Ben Philpott at UNC, where he looked at a couple of thousand drugs, and actually he used the mice that we developed while I was at Baylor College of Medicine, and so that's the tool that we're going to use at Texas A&M. And so we're simply going to take drugs that have been approved by the FDA, drugs that are in the process of clinical trials, and even small molecule compounds that have not been approved for use, and Dr. Weaver's going to talk about this too as well. And we're going to see, not knowing what any of these guys do, whether or not we can reactivate that paternal allele. And so we have some unique equipment at Texas A&M. We're going to be working with folks down at the medical center where we can literally screen thousands, if not tens or hundreds of thousands of drugs very rapidly to see whether or not they will reactivate this paternal allele. And after that's done, if we do find that, and I think there's a really good chance that we will, given what's already been done. I wouldn't say that we're reinventing the wheel, we're just adding to the effort. But once we find that, then we have the expertise here to start testing those drugs in mouse models to see whether or not they can improve neurological function. And so our hypothesis, our reasoning is that reactivating that allele from the father that's normally silent may produce protein in the brain, and then it may compensate for the loss on the maternal chromosome and restore some function of the brain. We don't know what that's going to be. There's a lot of problems with delivery systems and what regions of the brain this will happen in, but that's the thinking nonetheless. The last component of this project is our involvement is, and Becky mentioned this too as well, so there are patients that clinically have been diagnosed with Angelman syndrome, but when the genetic testing has been done, they come up negative for everything. And so there's a battery of tests that are run through various genetic laboratories that are somewhat common, and so they look there first, and then if they don't find anything, then there's not a lot of research that's done in those laboratories. So you either have to find a lab that's willing to look at other genes and other mutations, or you're really left with nothing. And so what we're going to do is we have some other unique technology, and things have really advanced in genetics in the past couple of years. And so instead of looking for specific mutations that exist in the genome one by one, what we can do is we can sequence the entire genome in one reaction. And so we can determine all the variation, all the genetic variation, all the mutations in one's genome in one experiment. It's somewhat expensive. It's very computationally extensive. The computers that we use to do this are supercomputers, but we can do it nonetheless. And the cost is, I say it's expensive because it's more so than a conventional genetic test. But when the Human Genome Sequence, or the Human Genome Project sequenced the human genome, that was, what, $3 billion, I think? Now we can do it for about $8,000 or $9,000. And the turnaround time is about a month. So things have improved dramatically in that area. And so we'll be looking at individuals that have been diagnosed with Angelman syndrome. We'll be sequencing their genomes or sequencing parts of their genomes and using other genetic tests to try to identify the molecular lesions that are causing their condition. And what that may do for us, though, is not just a diagnostics tool, but we suspect that there may be other genes that are mutated that are giving rise to these Angelman-like phenotypes. And this has been reported. There's at least 3 disorders that I know of: Rett syndrome, Pitt-Hopkins syndrome, and 2q deletion syndrome, that are oftentimes misdiagnosed as Angelman syndrome. And as scientists, what we think is that the reason that's probably happened is because those genes are probably involved in the pathways that the UB3A gene is involved in. So it teaches us a lot about what UB3A does in the brain and what other genes are that may regulate that. So in some instances, we may find just novel mutations in known regions, but in other cases, we may find new mutations in other genes that may glean information on what UB3A does in the brain. And so that's what we're going to be doing. We're going to be applying some fairly advanced technology and trying to pick apart these lesions that are also causing Angelman syndrome that are not detected. So in a nutshell, that's what we're going to be doing. And we're going to be working with these guys and with Ann to, every time we find something, if we find a drug, then, like as Dr. Weaver mentioned, we're not going to wait for it to be published. It'll immediately go to him, and he can start testing these things. And so we're going to try to shorten that time that it takes between discovery and translation. So that'll be our role in this project.
I just wanted to add to that really quickly. I know there are some families in the room who have clinical diagnoses. Make sure we know who you are, because this applies to you, right? Because we may be able to help you figure out what the clinical diagnosis is for your child and why they have Angelman-like syndrome. I just want to remind you, too, that when I gave you your timeline, that they had found that deletions on chromosome 15 caused Angelman syndrome, and we didn't know why that worked. There were a whole lot of clinical cases out there for Angelman syndrome who didn't have deletions, and we found out that those were mutations in UBE3A, and that's what got us on this pathway, right? So the individuals who have UBE3A mutations are what started us on this path to say that there are therapeutics out there. So imagine there's someone out there with a clinical diagnosis of Angelman syndrome who has a mutation in the most important gene that UBE3A regulates. We have a whole new way to look for therapeutics, and they might be very specific, and they might not have things like toxicity associated with them. So, you know, we're looking for Scott to find some miracles for us here. but you know, the clinical cases, let us know who you are so we can let you know when we're ready to start this kind of stuff, and help us find others who are clinically diagnosed who may not be as active in our community, because we need them.
Thanks. I think that's great. So you can start to see where the synergy lies here. Okay, so we have a cutting edge technologies with both of these guys that have this capability in their laboratory. The work that's ongoing in my laboratory is manyfold. We're continuing to do the same kind of drug research that Scott was talking about earlier, using the mouse model that he actually developed. So he did this very kind of cool thing where he put a fluorescent protein on the backside of the paternal gene. So if the paternal gene starts being produced, you can actually see the neurons will glow yellow under fluorescent light. So it's really a beautiful, elegant system in order to do these drug discoveries. So we're looking at this as well. Anything that he finds, we can verify in our laboratory. Anything that I find, he can verify in his laboratory. So you can see where that synergism and that immediate information dissemination can be truly beneficial. Any drug that we potentially find, what we can do is we can send it down to Dave, who's developing, and you have to understand, making a neuron out of a stem cell or out of a blood cell in a petri dish is, you know, this technology didn't exist just a few years ago. So we can look at this in actual human neurons and say, are we seeing the same effect of the drug? Now we'll have new animal models to test things like toxicity, side effects, effectiveness in any of these new drugs that are developed. So now you can start to see how all this is really starting to come together. The other aspect of these drugs is not only do they turn on the paternal gene, or can they be effective therapeutics? I mean, we're looking right now at a number of different FDA approved therapeutics that could be potential therapeutics for Angelman syndrome. We're actually working with a couple drug companies that they're not giving the drug out to anybody except our laboratory, so that we can look at this in the animal model. But one aspect of this is in the seizure control, something that is really kind of, you know, not really looked at very hard in the AS field. And so if this was Anne sitting over here, I'd be pointing to her, and I could say, Anne's going to take this. She can do EEG directly in the mouse model. Okay, she can look at things like sleep. She can look at different activities. She can look at how those EEGs change over time. So she's looking at it in the eye of a epileptologist, and can evaluate all these drugs as well. So some of the drugs that we're looking at in my laboratory have anti-seizure properties, but we think that they're also going to be beneficial in Angelman syndrome as well. So the research that I do, I mean, I'm looking, you know, not on genes or transcription factors. I'm kind of looking at the level of the neuron and how neurons interact with each other and how they communicate with each other. And we know from the mouse model that communication is disrupted. Those neurons just don't communicate with each other. And if you've got a disruption with two neurons that can't speak to each other very well, they don't speak to each other very well. But if that's happening everywhere in the brain, then you have a syndrome. You have something like Angelman syndrome. So in my laboratory, we're trying to figure out how we can increase that communication. If we can increase it a little bit across a few neurons, what can happen if we can do this globally? Can we raise up the entire function of the brain and overcome some of these defects that we see in the AS mouse model? We're looking also on drug discovery for cognitive enhancers, and this could be applicable to a lot of different disorders, but one of the things that we're specifically looking at is cognitive enhancers in the Angelman syndrome mouse model. Can we increase their cognitive ability? And we're not looking at this in little tiny mice. We're doing all this in adult. So I think that it's important to know that we're We're looking at these changes in mice that have had Angelman syndrome their entire life, and they're not kid mice, they're adult mice. So if we can see changes there, we're hoping that they can translate into adults of any age. So that's in a nutshell, that's what the grant is doing. We can easily work with each other and use our own expertises and our own specialties to really drive the science faster than we could ever do in our own laboratories by ourselves. And having this kind of synergy, you can kind of see where real-time information dissemination is just, it's incredibly important. Plus, you know, we all have a different perspective. You know, geneticists are weird. Geneticists think of things much differently than biochemists do, or you know, in the realm that I live in as well. So getting his perspective on things has really changed how I view my own science. They really are weird. He's smiling over there, but they really are weird. Yeah. He's going, yeah, those neuroscientists are funny guys.
They're trying to figure out what's wrong with themselves, right?
So Scott came and he actually sat on the committee of my graduate student who graduated a few months ago and is off pulling Angelman syndrome research into another laboratory across the country. But so he came over and we sat and literally talked science for, about 18 hours straight. It was really incredible, and that kind of interaction and synergism, it's hard to explain how much that elevates the ideas and thinking outside of the box you can have when you're thinking about something as complicated as Angelman syndrome. So I think with that, we'll just kind of open it up to whatever questions. I've said this before, there are no stupid questions, only stupid answers. So we'll try to keep our answers as smart as possible, but feel free to ask anything that you want. I mean, this is the opportunity to get up and ask us pretty much anything. And if it has to do with genetics, then I'm going to send it over to Scott. But if you have specifics about what they're doing, how they're doing, what they're thinking, you know, this is the time to ask them.
And we have lots of questions from online, so we're going to go back and forth. So definitely line up at the mics because we're filming it and people at home can't hear your questions if you're not at the mics. I'm really loud, but most people aren't. So, but you go first.
Okay. Just a question for Scott. You'd said when you were sharing the information, you weren't going to publish it first. Does that have an impact for the wider scientific community as to how it's viewed if it isn't published, or did you just mean that you would share it while you're going to publishing the data?
Yeah. So I didn't mean to imply that we wouldn't publish it or we would wait to publish it. I think that the comment was made that because the way the system is set up, for others to find, you know, to get that information, it usually comes through the literature. And that can be a long process, you know,
Sure.
writing the manuscript, having the manuscript peer reviewed, addressing any issues that the reviewers might have, and then actually making it online or in a journal. And so what I meant was that if we on, you know, a Monday we find some exciting compound, then by Monday afternoon Ed will know about it. Okay. And he can, you know, gear up to potentially look at that. And so what we're trying to do is really cut the time that it takes to move this information around.
Okay.
But it'll still go out and be peer reviewed and under the same rigor that it normally is for any other project that you might have.
Okay. All right, thank you.
Hi, thank you so much for being here and for all the work that you're doing, first off. And secondly, my son has UPD, he's 4, and we found out last year he was born premature. And I think really the first sign, just FYI, was that he smiled for real when he was 34 weeks old. He was born at 31 weeks. And I thought that was a real smile, that wasn't gas. And I was kind of, you know, I found that interesting. But anyways, if you turn on the paternal allele in a UPD, would that turn on both? And would that have any implications that are negative?
The question would be if both alleles are turned on. So there's another condition, and we actually study that in my lab. It's called 15q duplication syndrome, where there's an excess of UB3A. And that causes autistic-like phenotypes. There's mild developmental delay, and then they also see seizures in that condition as well. And the severity of 15q duplication, it increases with the number of copies. So there is something about, going back to my comment, there's something about this gene, and it needs to be very tightly regulated. And we're trying to understand what that is and how that works. But I think that having not enough is much worse than having too much. And we don't know that for certain, but if you compare the two conditions, I think that might be true. And so there may be some side effects from that. Maybe I wouldn't say side effects, but maybe some new phenotypes or new traits. But the hope would be that it would improve the other deficiencies. And so it would be different maybe, but not worse.
Or what if there's a drug that only turns the paternal gene on 50%? I mean, you never know, what the future is going to hold. But in that case, you would, the UPD would have 100% of the protein that's necessary. So, yeah, it's tough to know.
And a follow-up question, if I may. Is it possible to maybe, is it possible that some of the clinical AS patients, or maybe some of the patients that have autism, like a low-functioning autism or a high-functioning autism, may actually be clinical AS patients? I mean, is there some sort of potential crossover with that?
Can't remember the question now. My ears are still ringing. There may be. I think that I suspect that there are probably lots of variants that exist within UB3A or environmental factors that dysregulate UB3A that lead to some mild version of AS or autism. Yes, I believe that's probably the case.
Do you think that there's going to be some partnership with that later on or soon?
I think so. I suspect so. Dr. Baudet had looked at that for years. When I was there, they was looking at UB3A as being a major player in autism, that didn't have Angelman-like or not phenotypes, but I think there's a lot of people that would believe that may be the case. Yes.
Thank you.
Okay, over here.
Hi, my name is Eddie. At the beginning, Becky said that AS and Alzheimer had like a close relationship. I would like to know what it is.
What did I say?
Yeah.
Autism
as
and
far
AS
as a
has a
close
relationship.
like
That's what I understood.
they're not really married, but so
Or related.
yeah, so there are some reports out there that UBE3A, at least the protein, is decreased during age, and decreased even more great in some models of Alzheimer's disease. So if you think about UBE3A being a protein that's essential for normal learning and memory, I mean, this is something that there may actually be a connection between those two, but we don't know for sure. I mean, that's. I would love to marry those two things together. I would love to marry Angelman syndrome with any disorder that affects millions of people for the very reasons we were talking about earlier. But we just don't know for sure. I think the biggest problem is that for UBE3A, it is believed to be a ubiquitin ligase that is a housekeeping gene. But I think what we know now is that UB3A as a protein, and this is coming from the view of a biochemist, is doing much more than that. So it's probably doing things in the cell and affecting genes and affecting the biology and biochemistry in the neuron that we haven't even scratched the surface yet. So there really should be no reason for this type of protein and this type of gene to be missing and cause such a varied symptomology in the human. I'm getting louder and louder.
Yeah.
That's good. Okay, I'm going to beg Aaron to give
Thank
me
you.
a moment and let me ask a few questions from online. One of the ones that we have from online is, can Dr. Dindot tell us how his research compares to that of Dr. Philpott?
So I think that there's a lot of overlap actually in what we're doing. I think we're both, I know Dr. Philpott, I would say well, and consider him a friend, and I think we're doing complementary research. He's clearly been working this field for some time and doing very similar things that we are, and the hope is that we'll take different perspectives and different approaches to get at the same goal. I mean, I think that one thing that's unique about what we've been doing is trying to understand the mechanism. And so I think that information from that project will hopefully open doors for new therapeutics that we just don't know about. But I think they're complementary.
And Scott's being really, really modest about this, but the work that Ben Philpott did was, I believe, with the mouse that you made. Is that not correct? Yeah, so it was Scott's mouse that allowed Ben to do his work. So this is how the researchers actually collaborate with one another and do help, and we just want to speed that process up and allow them to do it in real time. Erin.
Hi. Well, I'm very excited by the research into more mechanisms that could be causing Angelman syndrome and by studying our kids with clinical diagnoses. My daughter has a deletion, but I know so many families with a clinical diagnosis, and it just seems like I'm wondering if you think you'll find more mechanisms that could be causing Angelman by studying our kids with a clinical diagnosis, or if you think you're going to find more related syndromes, but not ones that actually directly are mechanisms causing AS in the clinical kids. Does that make sense?
Yes, I think it's and I'm not a clinical geneticist, and I think someone like Dr. Bodette is more qualified to answer these types of questions. But you could imagine new conditions being named that have very similar phenotypes. That's one thing that could happen. But regardless, my interest and my thinking about it is that it will reveal potentially novel pathways that this gene is involved in, or how it's regulated. So, for example, Rett syndrome is the protein that causes Rett syndrome is a methyl binding protein, and we know that this mechanism of regulating UBE3A involves DNA methylation. And so, for years, we've all thought that the Rett gene may be somehow regulating the Angelman gene, and so that opens up doors for that. The same thing for 2q deletion syndrome, where it's a methyl binding domain protein, and so we think possibly the same thing. So I think that's the sort of doors that will be opened, that we may learn, this, I think Becky mentioned this, that maybe this gene is critical. It's one individual that's maybe has a mutation in that, but it really just advances us years in terms of what's going on and what this process involves.
Well, I think as families, we can go out there and find the clinical diagnoses. We know them. We can make a database today on Facebook, So.
And hopefully this is okay to say there is a Facebook group for clinical cases for Angelman syndrome. You need to contact Yvonne Hamrick if you want to be in that group, and that's another way that we can make sure that we find you and that you get support as well. Oh, can I do an online question, or do you have something?
Yeah, I just want to add something really quick.
And I've got, one more question. Go ahead.
The other thing I wanted to say was that, looking at this not just in the aspect of the genetics, but also of the biochemistry. So when you think about biochemistry in the neuron, there are proteins that are kind of lined up. One affects another, that affects another, that affects another. And those strings of proteins are essential for the neuron to work properly. So when we talk about things that are kind of upstream or downstream from UBE3A, since we're not really sure what UBE3A is doing, if it's in that particular chain, what we may find is proteins that are upstream or downstream of UBE3A that have never been known. Now, there's a lot known about the proteins that are in the neuron. There's a lot of proteins that are targeted with particular therapeutics. So, we could potentially be finding a protein that a therapeutic has already been developed for that we just don't know about. Mm-hmm. So if we were to find novel genes, we could also make novel mouse models as well to look at particular therapeutics. So really, it's limitless what we can do if we can find these other genes that could be associated with the clinical diagnosis.
And I think as families, we need to find the families who have been denied a clinical diagnosis, because I found that's very subjective, right? Some doctors will quickly give a clinical diagnosis and others will say absolutely not. But I know kids who, ended up having a positive molecular diagnosis of Angelman who were repeatedly denied the test, saying they didn't meet the clinical profile. So I think we really need to find the families who have sought it, couldn't get it, and still take a look at their genes too.
Absolutely. Yes, because I mean, as more and more individuals are being diagnosed with clear molecular cases of Angelman syndrome, we're finding kids out there that talk in complete sentences, that can write, that can do all kinds of things because they don't have the fine motor issues or the gross motor issues that some of our other kids have. They're never going to get a diagnosis of Angelman syndrome because our kids don't talk, right? And there are some people sitting here in the room who can tell you their kids talk. So I think as we start diagnosing more and more, getting these and getting people in who've been denied clinical diagnoses, the spectrum and the picture of Angelman syndrome is going to change, and it's going to be much, much more than 1 in 15,000. I think our community's much bigger than we understand,
right,
and we need to get them in.
So I also have a question about the paternal allele and activating that. Maggie's original diagnosis was Prader-Willi, and I know that's quite common for a lot of us with kids with deletions. I have yet to find a physician who wants to look at the overlap between Prader-Willi and Angelman. But it concerns me when we talk about activating the paternal allele because we already have a lot of crossover symptoms, and I think we just really need to understand that. So I would love to recruit someone who would study that. But there's so many of our kids. So my daughter has osteopenia. So do kids with Prader-Willi. My kid has a very abnormal appetite. So do kids with Prader-Willi. Many of our kids have insatiable appetite, and Dr. Oliver has already documented that. We've got a lot of kids with unusual puberty. So do many kids with, that's a clinical characteristic of Prader-Willi. A lot of our kids have delayed growth when they're very young and they grow very small. Our kids have unusual metabolism. Unlike kids with Prader-Willi where they have a very slow metabolism, our kids often start out very rapid, but then it becomes abnormally slow as they age. So I'd really like to see if we're studying any of that and how even with a deletion, if there's something going on with the paternal allele or something going on in that area that could be impacted by activating the paternal.
Any of you want to talk about
that? And so could you repeat the question though? I mean, is it that?
Sorry. Well, you mentioned.
I think what she's saying is that the kids with Prader-Willi and the kids with Angelman syndrome do have some overlap in their symptomology,
right,
even though the deletion is on the opposite chromosome or the mutation is on the opposite chromosome. So the question is there something behind that overlap?
And kids with UPD and kids with imprint center mutations often have the most overlap. And we see, for example, OCD in kids who already have 2 paternal alleles. So to me, when you stated it's a milder symptom because of the 15q duplication syndrome, so it's probably not that big. It's not as bad a deal if we activate the paternal allele and see some side effects from that. But there's also Prader-Willi, and we're already seeing, for kids who already have 2 paternal genes, we're already seeing symptoms of Prader-Willi. So what are the implications for that with our research?
I think all I have here is a best guess, and those 2 chromosomes are so. Deletions on the paternal chromosome obviously give rise to Prader-Willi. It's a different gene though, but the similarity is that it's a common deletion. It's a large deletion. If it's paternally inherited, it takes out the SNO RNA that causes Prader-Willi. If it's maternally inherited, then it takes out UB3A and causes Angelman syndrome. But what we do know is that those 2 chromosomes are, like I mentioned, it's more than just those 2 genes. It's a very complex region. And so my best guess is that in some certain molecular lesions, it's possible that the other chromosome can be affected. And so that's why it's giving rise to phenotypes that are similar to Prader-Willi or possibly even vice versa. And so that would be my best guess. And that may be what's going on because we don't know how this region really works, and it's very likely that they need to both be normal to function normally, and so mutating one chromosome may have an impact on the other. And so that may be the reason for that. But again, I'm speculating here. But
it'd be exciting to find a Prader-Willi researcher to give some insight into that as you guys start seeing things in your models, to have someone who actually specializes in Prader-Willi take a look at that. I've yet to see anything where someone with Prader-Willi expertise takes a look at Angelman, and I think that's long overdue.
Well, I think there's some groups that, I know Dr. Bodette had an interest in Prader-Willi syndrome, and they were studying that. And so I think they do exist, and I think that, like in my lab, we We don't have an active program in Prader-Willi syndrome, but we're clearly thinking about that gene all the time. You can't, they're part and parcel. You can't think about UB3A without thinking about all those other genes there. So clearly anything that we might find could have implications for Prader-Willi syndrome. Absolutely.
Okay, hold on just a second. Let me make sure we get some of the online questions as well. We have a question for Dr. Weber about both secretin and CAMK2. And so do you think your work on secretin can help figure out delivery for therapeutics? And are you continuing your work on CAMK2 in relation to Angelman syndrome?
Yeah, so yeah, secretin is a, wow. I wish I had a blackboard. I wish I could just impart all my knowledge onto you guys with some kind of mechanism. Secretin is a protein that we've worked on through collaboration with another scientist. Secretin is it's a specific protein that's been associated with autism, and the question about CAMK2, I mean, clearly, if you can rescue the phenotype of the AS mouse model with a CAMK2 mutation, CAMK2 is involved somehow. So yes, we're not doing so much work with the secretin anymore, but we are focusing on looking at CAMK2. And I think any of these therapeutics that we have, I mean, CAMK2 is one of the first things that we look at is CAMK2 changing, because we already know that if you can change CAMK2 in a specific way, you can alter how severe the phenotype or the symptoms are in the mouse model. So, yes, more peripherally than directly looking at CAMK2, but we're continuing that research.
Okay, so we'll come here and then we'll come back. Okay, great.
Hi, it's great to see you. My name's Susan Hogarth. I'm from Colorado. I have a 7-year-old Angelman boy. And I think I piggyback off of the clinical. He's a UB3A mutation. So he was, of course, for the first 2 years of his life, through all the doctors, just delayed because he didn't have seizures. And so at the Children's Hospital in Denver, Colorado, they just said, you don't need to do genetic testing, just give it some time. So read Colin Farrell's first article about his son in 2007, looked at that, said he looks just like Jake, and went and ourselves diagnosed him, went back to children and said, we think. And they said, well, we'll test that on the Angelman Syndrome, the 2 phases, and just work on that first, because we had another little girl come in and she was high functioning as well. And of course he passed the first because he's not severe, and then failed the second. So he has a 7 base pair missing on the UB3A. And so clinical diagnosed, because taken from the blood type of both the father and the mother of ourselves, that was not clear, but based on the characteristics that we provided and the UB3A piece sliver that was missing, they diagnosed him as Angelman. So I've always felt it'd be great to somehow talk to you about him. I would love to, provide information about him, thinking that he might help in some way. And I'm just new into the clinical studies. He hasn't been in a study at all. So I guess that's my question is, and maybe Becky just brought up about this Facebook link of these kind of kids that are in this second phase, these more non-deletion type of individuals, and how they can be released to you, or is there something, I'd love to bring him to you, I'd love to show what his genetics are, just in some way to maybe if he can bring something to the table. And so I guess maybe that's my question, is how to get involved in some of those clinical studies. That's something we haven't done yet. So.
This is for the clinical trial? Or just in general?
Just in general, anything. Are you in an active studies? I'm not in anything yet. We haven't done any type of studies. I haven't entered him into anything yet. So. This was just a new thing. I keep thinking I need to get him into something. So.
Yeah, so I think that you never know where the next discovery is going to come from.
Exactly.
And if there's, Dave talked about this earlier, getting communication from you and emails from you just about who you are and what you're doing has an effect. But also, but also it's.
I think it's recording from it, so he's telling me.
Oh, it's recording. Sorry. No problem. But I think it's also important that we don't really know where the next discovery is going to be made. So, I think contact any of us with those details, and if it's, is in the eyes of a geneticist, if this is really something interesting and can give us insight on what UBE3A is doing, then I think it's extremely important. We get a lot of information from the parents. The information from the parents about their kids have influenced my research. So, no, that kind of interaction at any kind of level I think is important. So, yeah.
So
contact
how would we
me. Send your information to me.
So how would I do that? Like, how do I get a hold of any of you, I guess? Or
go to weberlab.com.
Just do that. Okay. Okay. So
Google Ed Weber, and you will find or Scott or Dave.
So just directly we can do that?
Sure.
Oh, okay.
Yeah.
And then the Facebook, Becky, what was that again?
For clinical cases? But your son has a deletion in UBE3A, right? A small one?
A small 7-base pair, yeah.
Is there a 3-base pair mutation or 7?
7-base pair on UBE3A.
And it's and then they told you it's at the end?
No, it's within the middle. It's a different.
Oh, we've talked, right? It's in the intron.
I think it's in the yeah.
Yeah. Okay. So clinical cases, you should look for Yvonne Hamrick on Facebook for that group.
Okay.
The other thing that you could potentially do is contact someone at the Rare Disease Clinical Research Network, the RDCRN. If you send me an email, I can get you in touch with them.
Okay.
But they run the natural history studies at all the various different sites, Boston, San Diego, Texas, Greenwood, and I think you could talk to them about whether or not they're interested in starting to look at clinical cases for the natural history study. I think it's something that we're going to have to start doing because we want to find people with clinical cases, but that would be something to do as well.
Okay. Okay, thanks.
All right. I'm going to ask Stacy to bear with me for just a second so I can ask one more question from online. I'm getting multiple questions asking if Ed could please give some sort of update about the clinical trial and what's going to happen in the future with potentially expanding the clinical trial. I know we talked about it last night, but I think a lot of people have missed it online. So I'm going to ask him if he could just repeat the statements that he made last night so people online can hear it if they missed it.
Okay, so as far as the clinical trial, Joker goes right here. We're finishing up the second half of the clinical trial. So the way that this was designed was with the 24 patients, it would be split into 2 because we can't do all 24 at one time. So it was spread over time, and this is why it took as long as it did. From what we can determine from the initial 12, there appears to be at least some positive effects in different areas. We don't know if that will hold true through all 24. This is why it's really important just to wait until we finish the clinical trial, and I can't really say anything about specifics because of that. Again, it's kind of like reading the first half of the book. I really don't know how it's going to end. So we have to wait till the end of that. Now, the Rare Disease Clinical Research Network, the RDCRN, this is a multi-site group of clinicians that are there to help with this kind of research. So we can do the footwork on this, and we can develop these therapeutics, and they're really the ones that can continue this on and do it in a way where it's easier for patients to go to those specific sites instead of flying down to one site. So I think that they're waiting to see what the entire clinical trial is going to look like when we can get all of the data together and present it to them. Of course, they're not going to make a decision that's going to be extremely costly and in man hours and in money before they see all of the data in front of them. And that's, surely you understand that's what has to be done before the expansion of a clinical trial. But what they have said is that they're willing to be able to facilitate this. If everything holds out, and we assume that it will, but if everything holds out, then they're saying, yes, we will help expand this, and it will be an alternative for parents to go out and really, what they can do is they can really help determine whether this is going to be an effective therapeutic or not by doing other things with the clinical trial and working through that network of institutions. So Yeah, I'm sorry that I can't say specifics. I would love to, but it would be premature for me to say anything. But I think that it is important to know that we're extremely fortunate to have the RDCRN there, and extremely fortunate for them to say, yes, go register with us. We can facilitate this as quickly as possible if need be, and it gives all of you an alternative to rushing out and doing this with your pediatrician, because there's still a number of questions that we still need to answer as far as the minocycline goes. So I would just say, again, be patient, and register with the RDCRN, and then wait for something to happen there. Becky?
Now I get up here and I'm like, okay, what was I going to say again? So I'm Stacy, and I have 3 kids with Angelman Syndrome. They're UBE3A. We were one of those families who have been turned away and argued with for 7 months to test my daughter because she didn't meet all of the criteria. Anyway, so it was more of a statement, maybe not a question. My last 2 children, I got their stem cell blood cord and kept that. And so I've offered that to some other people who I know were doing stem cell testing. So just to let you guys know that if that's something that would shorten time in creating, getting stem cells and, turning them into something else, to find that there's probably parents out there like me who have that, who would be willing to donate it, to see if it could speed up, or something, the effects that you guys are, having to change this into that before you get to where you, want to be. So.
Nope, just thanks for that kind of resource. It could be very helpful.
And that was actually my question too. My name's Amy Addison. I'm from Tampa, Florida, and I have an angel, Ethan. He's 10 years old. Yay, Tampa. And that was my question: is if there's any research out there using cord blood from a sibling, because I know cord blood has cured a lot for other things.
Yeah, I think at this point it's at too early a stage to know how they would be used in a therapeutic sense, aside from testing the therapeutics that we're thinking about now. But again, maybe having different kinds of samples would be helpful. So I think that's good information for us to keep track of, and I appreciate your volunteering that.
Thank you.
This question is actually from Harley, so I'm just going to read it the way she typed it. I need to know about how you test left side brain in the mouse for voice, because mice not speak.
Good question, Harley. I've been saying for a long time that Harley's a scientist, and I think that you can see that.
Yes, it's true. Mice do not speak. But they do communicate, and they do vocalize. And they actually, small mice, if you remove small mice from their mothers, they vocalize at an ultrasonic level, and there's actually equipment that can test that. But I think that the vocalization and verbalization are 2 really different things. So in the mouse, Angelman kids will vocalize very clearly, so it's really hard to be able to test that in the mouse model. We don't treat the right brain and the left right hemisphere and left hemisphere biasly with my mice. We use it all. So we haven't done specific tests on right side or left side, or really looked at that aspect in the mouse. This may be actually something that, because the rat brain is much more complicated and they have more specifics on their vocalization in when you that ultrasonic vocalization, and much more known about rats, we actually may find that the rat model will give us more information on that, and it actually could be something that we could test later on. But I think we're just looking at the basics now.
I mean, I would just add to that and say, you're right. I mean, these animal models are not always the best model of these diseases. So there are limitations to all these kinds of animal models, but they're the best tool that we have right now to test things like efficacy and toxicity and safety before we can even go into a clinical trial. But you're right, it's not exactly the same.
Okay, I'm going to let Gina talk, and then I want to do one from online, and then we'll come back. So, Gina.
Okay, my question is, Will, my son, is 4 years old. He is UPD. Could you tell us a little bit more about turning on the paternal copies in reference to voice?
Well, in turning on the paternal copy in boys, I think...
In voice. Voice. In reference to being able to speak.
To being able to speak, yeah. I mean, to the extent that the disease is caused by a deficiency in UBE3A, the hope is that if we can reactivate that we should be able to rescue many of the things that protein does. And it's not completely clear how the deficiency in UBE3A prevents speech, but it's hoped that if that protein were to be turned back on, then it would allow learning to a point where they could develop speech again. I don't know if the defects in UBE3A alone contribute to all of the spectrum of different symptoms that you see in Angelman syndrome, but we believe that it contributes to a large majority of it, and especially things like learning and speaking and spoken language. So the hope is that would all improve.
Okay, so if they can speak 20-some words already, you think that they could learn and develop speech if that works?
If we can turn the gene back on, it should be better. That's what we expect.
Okay, thank you.
Okay, we have a question from online that I think is important. Are there any thoughts on any detriments or potential dangers to turning on the paternal allele? So do we know if we're going to potentially do something negative or cause other catastrophic problems by turning on paternal that we haven't maybe talked about or thought about?
Well, I would just repeat something that Scott already said, which is that I think too much expression of this gene could have a potential drawback. I think that's a lesson that the gene therapy community is learning in general. I think and for many types of genetic diseases where gene therapy has been applied, it was so hard in the beginning, and it's still hard, to introduce or activate a gene that, in the beginning, the thought was, well, if we could just turn it on as high as it can be turned on for as long as it can be turned on, that's a good thing. And we're finding that that's not always a good thing, that often these genes are carefully regulated in how much gets expressed, and too little is bad, and sometimes too much is bad. Having said that, I think the therapies that we're contemplating would be things that can be regulated, like a drug therapy. You could give more, you could give a little bit less if it gets turned on too much. Hopefully with the kinds of therapeutics that I'd be working on, it would be similar. So there should be some level of regulation, but I think that it does need to be considered. So I think what Scott mentioned before is that there are some children that have duplications of this region, and the thought is that they have more UBE3A turned on, and sometimes that's been associated with autism. So there's a concern about that, but I think with the therapeutic types of approaches that we're considering, that's part of the design, that we can hopefully regulate that as well.
So I'd just like to add one thing to that, and that is I think that speaks to the importance of model systems and having more of them to be able to test these things. So Again, it's not, mice aren't humans, and we all know that, but it at least gives us a tool to test these. So if there is some effect, in theory, we should be able to see that in a mouse model. And so, hopefully, we will look for these things. We don't know how things will unfold, but we do have the tools to attempt to determine whether or not reactivating that is bad. And we just don't know the answer to that yet.
Has this been done with any other disorders? Has anybody ever reactivated a gene or turned a gene on or increased its expression?
Well, I would just, so in terms of gene therapies have been attempted for things like Fragile X syndrome, where we know what the gene is there. Also in Rett syndrome, there have been models where the MECP2 gene that's mutated in that syndrome have been restored in animal models. And that's what gives us the insight that sometimes overexpression of these genes can be problematic. So, that kind of research has led us to understand better about the specific cases that we're interested in UB3A.
Okay.
Hi, my name is Sebastian Moniz. Was diagnosed with AS back at Europe. And my brother's question is, what is the chance of their second child being diagnosed also with AS? Is it completely at random, or is it depends on their family genes? Is it related to their past family genes, or is it what's the chance that their second child will be diagnosed with AS?
So this is your child?
No, this is my brother's child.
Your brother's
my
child.
niece, yes.
Do you know what the molecular diagnosis is for your brother's child?
It is a deletion.
Okay, so deletions occur about 1 in 15,000 births.
Mm-hmm.
So the chance of them having another child with a deletion is about 1 in 15,000. So with a deletion, there's no great chance that you're going to have another child with a deletion because it's a random event. There's one small exception to that, and that is if your brother or his partner have something called a Robertsonian translocation. It's incredibly rare. But if they would it's in terms of family planning, and I'll just say I'm not a molecular geneticist, so they should talk to a geneticist for family planning. They should both have their chromosomes tested to make sure that there's nothing wrong translocation-wise, and then their chance of having another child with a deletion is almost nil.
And one more question, like families with 3 children that are diagnosed with AS, how's that happen?
So those children have mutations in the UBE3A gene, so they have actual physical changes in the DNA. So the deletions that occur are random. They're random events that occur when DNA replicates itself to make more of itself. And so that is a random event that happens. If you have a mutation in UBE3A, that's permanently genetically in the DNA. It's not something that's random. And so in that case, you have a 50% chance of passing that on to your next child. So.
Okay, thank you.
You're welcome. If anybody else has questions about that, Aaron Sheldon and I wrote 2 documents that are on the AS website under What is AS. One's called Testing 101 and one's called Genetics 101. I still think they're valuable, so if you would like to go read those, I think that can help clear up some of those questions. But definitely talk to your molecular geneticist if you're thinking about family planning, and don't leave until they explain it to you in a way that makes sense. And make sure that they talk to you in a way that makes sense. But if you do have questions about it, the community and I are happy to try to help you understand the testing results or anything else that you've heard from them that maybe didn't make sense to you when you were sitting there, maybe not listening. So, as always, ask. We'll help. Over here.
Hi, my name is Eddie. I'm from Guatemala. I have a 1-year-old baby recently diagnosed with AS, and we did the CMA test on him. I don't know if Dr. Scott talked about a single test that can show the whole chromosome, the whole DNA on a cell. If you were talking about the CMA test, chromosomic microarray, I think.
Well, I can't speak to exactly how the tests are run nowadays, and I've talked to some parents, and I think there's sort of an order that they go through where they look at the most common ones. But my point was that the technology is such nowadays, and it's making its way into clinical genetic testing laboratories. There's a few in the country that are currently doing this, but the technology allows you to, for the CMA analysis, to look at deletions and duplications throughout the entire genome in one experiment instead of just being focused on a particular region or a particular gene. And then in addition to that, whole genome sequencing, and more commonly what's being used now is exome sequencing, you can look for mutations in all of the coding portions of the genome in one experiment. I mean, it's a very large experiment, but it is one test. But those are just now coming out, and there are only a few places that I'm aware of that are offering those as clinical tests.
Mm-hmm. The thing is that when I got the results from the CMA, they told me obviously that there was a deletion in chromosome 15, which I also run the methylation to confirm AS, but they told me that there was an extra piece on chromosome 8. So I don't know if that might be important for your research to see more AS diagnosed, take the CMA test and see if that repeats on more of them.
Yeah, so there's definitely value in that information, and what's difficult though is that we all, presuming that we're normal, we all carry lots of, rearrangements in our genome, and many of them are benign. And so one thing that, because this technology is advanced, we're starting to see that there are lots of deletions and duplications that we have that it's unclear whether or not they're predisposing us to disease later in life, or that they're benign, so they'll have no problem. But then a lot of times when these rearrangements do occur, like say duplications and deletions, large deletions, there can be other regions of the genome that are affected. And in fact, you mentioned Robertsonian translocations, and so that may be what's going on as well. But that information is, in my opinion, it's valuable. So it would be nice to have that, and I don't know if there's a resource to put that, but all the information we have that we can get our hands on is...
It was done on Texas children, so you could get the information easily, I think.
Okay.
No, but I'm willing. We're willing to give it to you if you need it.
Yeah. So again, I'm not a clinical geneticist, and typically these cases are handled by board-certified clinical geneticists. And yes, the privacy issues are clearly there, but I don't know what the rules are. But I think if the parents willingly offer that information up, that should circumvent that. But I can't email Texas Children's and ask for that. No, I certainly can't do that.
Mm-hmm. Okay.
The only thing that I would add to that is just to kind of clarify, how that information might be used. So, the very best picture that we could get on how genetics relates to disease in general would be to have everybody's DNA sequence and to understand their whole health history. That way, we could start to make associations between the genetics that we see, and do any of those things, like this deviation on chromosome 8, do any of those start to reoccur in other people that have similar types of symptoms?
Yeah,
then we start to say, okay, this is associated. But when you have one case, it's hard to say with any certainty that that is having any effect. So the more kinds of genetic sequence that the community has to look at, along with very careful diagnostic of their condition, that kind of information is very helpful, and we build that person by person. So thank you for mentioning that.
Okay. I'm going to approach you later to give me my info. Okay. Thank you.
So a quick question from the online community. In the minocycline clinical trial, are you checking for seroconversion to positive anti-nuclear antibodies, or ANF? There was a Fragile X paper where patients had some positive ANFs at the end of the trial.
Joe, no. No, we're not testing for that.
Hi, my name's Elaine Pelly. We're from Madison, Wisconsin. This is our first foray into the Angelman's world, and it's been pretty overwhelming. But our son Ryan is 5, and the main reason I got up was actually in follow-up to that previous question. When Ryan was originally diagnosed, They told us, you know, it's most likely to be a deletion. We'll go looking for this, and when we got and they said, and lucky is that probably it's sporadic. Your chances of this happening again is 1 in 15,000 or whatever number they gave us at the time. But then they did Ryan's testing, and for reasons that were never really clear to me, they did markers on chromosome 15 and other chromosomes that might be likely to be problematic, and found that he had partial trisomy 13. And that's what got me my diagnosis of the balanced translocation that has been referred up there. So it's rare, but actually about 1 in 600 people have a balanced translocation. For it to happen to unfortunately fall in the area of chromosome 15 that causes Angelman syndrome would of course be less likely. But balanced translocations on their own are actually relatively common. So if you have gotten any clue that there is anything extra, that could be, that could be a translocation, which may not be as much relevant to your child, although it may be, depending on how big the extra material is, but certainly plays into your risk of future children being affected with Angelman syndrome. Certainly did for us. So that's just a statement, I guess. But also sort of a genetic question. I'm an endocrinologist, a clinical endocrinologist at University of Wisconsin. And so prior to any Angelman diagnosis, I was always sort of just interested in imprinting, because there's a lot of imprinting in endocrinology. And so I have particular interest in parathyroid disease, so pseudohypoparathyroidism and pseudohypoparathyroidism as an imprinting pair. And there's kind of, if you look at what we know about those two areas, there's some interesting sort of similarities in how UBE3A in that area sort of imprint. And I just wondered if there's much talk between people interested in imprinting in 15q11 and people interested in imprinting in other sort of imprinting diseases.
Yes, so I think that historically there have been, the imprinting crowd is relatively small, and that's one of the reasons why a lot of us come from, similar programs. I know that there have been efforts in the past to look at imprinting as sort of a general, area of science. My lab does that as well, and so there is a lot of overlap. We oftentimes look at how other imprinted genes are regulated to try to understand how another imprinted gene is regulated. And for the case for UB3A, it's one of the last on the list. I mean, the other genes that cause imprinting disorders, we know a lot more about them than we do UB3A. So there is overlap between those two. Now, how much is being done nationally and globally, I'm not sure about that, but those efforts are there.
Thanks.
My name is Vitaly Soloveitchik. I have a 2.5-year-old boy with Angelman syndrome. First of all, I want to just say thank you for Paul and Becky and whoever is. I think they're doing a tremendous job. It's an unbelievable job, and really appreciate it. And it's yeah. So the second statement is it's a great job to organize this, to bring you guys together, FIRE, and I think it's also unique, I guess, in medicine research, and it's really cool. So the first question is: are there any labs that work with other kind of models or branches research for Angelman syndrome that you would like to see in your group?
Yes, there are other labs. There are fruit fly labs that are using Drosophila to study Angelman syndrome. So I'm aware of a few of those. In my lab, we've done work with Drosophila too as well. We're not as active in that area, but Drosophila have their own, they're unique, and so they offer certain tools that you just don't have in mice. That some people, they're very powerful in terms of the genetics behind them. There are iPS cells labs working with, developing neurons from human cells, from stem cells. So I think there's quite a few labs. I think probably what's lacking right now is a larger than a mouse model. I think that's the only thing that I'm not aware of anyone working on. Although there is being a pig model developed for Prader-Willi syndrome. And so that has been done. And it's because the mouse isn't, there's problems with working with mice that have been engineered to have Prader-Willi syndrome, and the pig is thought to be a superior model for that. So I would say there's a pretty diverse group and diverse models that are being worked with.
I would also just say that you could think about research as being research aimed at trying to understand the disease better, and research aimed at trying to therapeutically intervene in the disease. And those are not mutually exclusive goals, of course. And the better you understand it, the more kinds of therapeutic targets you could try to approach. And in understanding what the disease is doing, you just know what you have to do better. But somebody actually has to try to make those therapies. Somebody actually has to research that part of it. And that's really what this FIRE team is about. We're not the only ones, but that's what we're going to try to do.
And second is easy question, I guess. The science is great, but show the money. Is any? I mean, for us as a parents, I guess it's what we're waiting for. It's magic pill, I guess. So, do you think is gene therapy is realistic in 5, 10, 20 years for cure or whatever, eliminate some symptoms? So gene therapy, clinical gene therapy for our kids, how excited we should be? Is it in horizon 5, 10, 20 years if you would bet?
That's the easy question?
Yeah. Yeah. I can say that we'll be a lot closer as we try. I mean, certainly gene therapy is something that's been investigated for maybe 2 decades. And we don't have any approved gene therapies today. So, I mean, that's kind of sobering. On the other hand, in the gene therapy community now, there's really a lot of excitement and a lot of very interesting things are happening. And there's also new tools coming online that we didn't have even 5 years ago. So I would say there's a lot of hope. I couldn't really put a number on it, of course, whether it be 5 years or 10 years. And how that's going to work, but I can only promise that I'm going to try.
Thanks so much. Thanks.
Can I just ask a question to Ed, just on the current minocycline trial? Are there other groups outside of Florida, and besides I think is it Germany, that are currently kind of trialing the minocycline or feeding information back into your laboratories?
So there are some other groups out there. They're not conducting what would be considered to be a clinical trial. I think that's the important aspect when you consider what's happening abroad. So yes, minocycline in Angelman syndrome, that's not exclusive to the clinical trial that's ongoing in Tampa or exclusive to this particular clinical trial. But I think that the measurements that we're trying to make is exclusive to what we're doing. So trying to understand all the different aspects that it could be affecting, that this is what makes a clinical trial so important. So, taking a child, giving them drugs, and seeing what happens is not the same as systematically doing it and then assessing them on dozens and dozens of different levels. So as far as feedback from any other groups that are out there, I know about it. I don't think that it would be easily integrated into what we're doing because of the way that we're doing it. So, I've heard reports, but I don't know any specifics about that.
Okay, thank you.
So we're going to get started again. We'll do some more Q&A. I know people have planes to catch and things like that, so we're starting to dwindle down. But we'll go for a little while longer if people have questions to ask. Head to the microphone, please, and let's start getting some questions. Don't be afraid. There are no questions that are problematic. Go ahead.
Hi, my name is Veronica, and I am an educator. And I just one of the things I really was impressed with the knowledge of the parents in this room is really, really amazing. So my question may seem really stupid, because you're probably thinking, oh, well, I looked that up on my second day of diagnosis. But listening to everything that I have learned today and what I've looked online, there are so many variables that could happen to cause the AS. Then why do they all have the common denominator as nonverbal speech?
Yeah, give it to the neuroscientist. So I think that this is actually a great question because it exemplifies what makes Angelman syndrome so unique. And it's unique in that it is only a single gene disorder. So in all of those things that were shown up earlier, in all the different except for the clinical diagnosed cases, but in all the known genetic molecular changes that we see that causes Angelman syndrome, it's always a disruption of the UBE3A gene. That's the common denominator. And we know that even just a mutation in that specific gene in one base pair that causes that gene not to function well will cause Angelman syndrome. So unlike severe autism, for example, where they're finding more and more genes, you've got identical twins that one has severe autism and one does not. Well, their genetics are identical. So, what kind of environmental factor pushed that child to have autism? That's not the case with Angelman syndrome. We can focus on that one gene, and we know that that one gene is the culprit. There's not going to be multiple other genes that are all working in concert along with environmental factors that could cause your child to have Angelman syndrome. So this is why, and I think this is why it makes it for a clearer goal for us to try to figure out how we can develop therapeutics and how we can target that specific gene. It all does boil down to the AS gene, UBE3A.
Thank you.
I'm serving as an interpreter for a couple that came here from France. The question is, you have talked a lot about the activation of the paternal gene, but as of now, the researcher Benjamin Philpott has finished his research on the activation of the paternal gene with positive conclusions. How come these results are not applied rather than looking in other places for other ways to activate the paternal gene?
Okay, so you can cure a headache in a lot of different ways, okay, and there's a lot of drugs out there to do that. And I think with the work that Ben Philpott did and the drug that he's using, which is a topotecan, this is a drug that has shown to turn on the paternal gene, and he's done it in a live animal. I think he's shown that he can do it in cells. Do you want me to stop and interpret?
Oh, that would be great if you
could
do
go ahead.
I'd love that. I just did what you already did, so you can keep going.
Okay, well, that was quick.
that works too.
Yeah, no pressure. So I think that it's important that we find new drugs that can have the same effect but show probably more specificity. And in the drug that Ben Philpott has found, it's a non-specific pharmaceutical drug that could be affecting a lot of other genes. So I don't want to discount from the immense scientific discovery that drug led to, and really it changed the way a lot of us thought about pharmaceuticals and about turning on the paternal gene, because clearly it can happen. The question is, can we find other drugs that may only activate that specific gene rather than trying to change a lot of different genes? And so I think it's important to continue on doing drug discovery.
And I'll just add one thing. I think that in science in general, I mean, you want, as many people as you can are working on a problem. I think this is through history, this is how science is, that's how we make progress. We don't just leave it to one lab and one person. We all work together in a lot of ways. So I think it's good that there are different labs, there are different ways of thinking about things that are trying to tackle this problem. I think that's good. That's not counterproductive. That's actually more productive, in my opinion.
Thank you.
I just want to thank you all for your work. My name is Rob Ciani. My wife and I, early on, we started taking our son to an acupuncturist, a nutritionist, and you know, it's obviously not a cure-all. But one of the things we followed Dr. Wen Han Tan's research with supplementation. You know, we haven't gotten too many results from that, but our acupuncturist had suggested acetylcholine. I don't know if you feel, I know that that's used in Alzheimer's and some other related disorders. I don't know if you have any feeling one way or the other. We haven't started it yet because, you know, I figured I'd ask an expert before I jumped in on anything.
I'm not comfortable in discounting any potential therapeutic or therapeutic design. Cholinesterases are the ones that are used for Alzheimer's disease and work quite effectively. Inhibitors, excuse me. So I think that when you're, if I answer this question in the role of a neuroscientist and electrophysiologist, that's saying if there's dysfunction in neuronal communication and how neurons communicate, I mean, they use neurotransmitters in order to do that, and different neurotransmitters in different areas of the brain. So this underlies the work that Wen Han is doing with the levodopa and increased dopaminergic, and there's been a couple papers that have come out on that actually even recently. There may be some relevance to other particular neurotransmitters that could be working in the brain as well. So I don't know if I would discount any of that. I've gone to an acupuncturist before as well, so I can tell you that for me it worked as well. So, you know, I don't really know. I would just say be careful. I would say that you need to talk to your pediatrician about any potential therapy that you want to go in, whether it be behavioral or something that's even slightly invasive like that. So, but I don't know of any research that would discount that, I guess is what I would say.
Thank you very much.
So we had one quick, I'll just come stand over here. We had one quick question online about fire, because I talked about it at the beginning, but the live feed wasn't running, and what it is that we're asking for in terms of the community's ability to help us with fire, which is the FAST integrative research environment. The two things we really need people to do are continuing to raise awareness the way we do as a community to make sure that people understand what Angelman syndrome is and to help us identify all the individuals out there that have Angelman syndrome so that we can help them both as a community and also scientifically. The second thing we need is we need research funds. We've been very honest and open about how expensive research costs. I will remind everybody that it's not because scientists make a lot of money, because we don't. It's a big joke when you're a graduate student how much ramen you can eat and still manage to have enough money for a beer. So we're not talking about paying exorbitant salaries. These are not people that are, you know, out living high on the hog on our money. And in fact, most scientists end up working 80 to 100-hour days anyway. We get the geek thing for a reason. So, but we do need the money because we need more of us geeks to be in the lab as much as possible to get the research done. And the more money we have, the more people we can hire, the more great scientists we can bring into FIRE, because there are others out there doing great research in Angelman syndrome that would benefit from this and would help us. So we're asking people, if you've got creative fundraisers or if you have donors you think would be interested in supporting FIRE, let us know. I'm happy to go sit and talk to anybody that wants to support us and tell them why we think they should. And sometimes we can even convince one of our scientists, even though they may not know we do this, to do something similar. So that was the answer from the question online, and so we'll go back to in the room.
Well, I think this related questions have been asked, but I think there's still a lot of questions about this, and this is maybe a little bit different way to look at it. But I guess my question is, in all the efforts, either thus far or future efforts, to try to activate transcription of the paternal UBE3A, are there specific plans to look at what that means for the other normally paternally expressed genes in that area? Because you have this one gene that we're kind of expecting to provide both the maternal and paternal role, or ideally, that's what seems to be what we're talking about. Any sort of concrete plans in maybe your future projects to both look at increasing expression of that, of the UB3A, but also what that does to the normal paternal role that chromosome is playing?
Yeah, so so one thing that's unique about UB3A is that it's at least that we know of, it's the only gene that is silent on the paternal chromosome. And so those, the other genes are already being expressed. And so I don't know that, it's not clear what impact it would have, but it's not, you wouldn't expect that we would turn them off. But we just don't know. The other thing to add is that we think that the mechanism is very different between the two. So there's a lot of unique things about UB3A, but studying imprinted genes, it is unique in that it's not regulated the same way that other imprinted genes are. And in fact, that's one of the reasons why my personal interests have gone into this area, because it's something that's very novel. And that's one of the reasons I think that it's actually, there's a possibility of reactivating. Clearly it can be reactivated. Ben showed that. And I think it's because of the unique way that it's regulated. But I don't, right now with the knowledge that I have, I don't think that that it will be a concern of the other genes on the paternal chromosome, but we'll test that. All these things we have to look at.
Good afternoon. My name is Heidi, and my daughter Claire is 14 months old. And I don't know if you can answer this or not yet, but with the clinical trial, if it moves on to a phase 3, do you think that they will open that up to the younger kids, or will it still be ages like 4 to 12?
I think that I don't know, but I can tell you this. As far as minocycline, there's not a lot known about the effects of minocycline, or at least the side effects or negative effects of minocycline in very, very young children. So, this was one of the things that we considered when we developed the clinical trial for the minocycline that's ongoing right now. I think that it would be advantageous to expand this to ages above 12, but I think that there's a lot of unknowns about minocycline in very, very young children. So I've done literature searches and looked pretty extensively, and I can't find any reports about the use of minocycline in very, very young children. But this is why we have internal review boards. So if I were to say, yeah, I want to give kids that are a year old minocycline, they may come back and go, that's pretty dangerous, and we don't think that you should. We just don't know. So I'm not exactly sure if there is an expansion of the clinical trial, and the RDCRN is involved in that. This is a group of really smart clinicians that know a lot about rare disorders and know a lot about these different drugs, and they would probably take all that into consideration. So I can't really tell you for sure, but I would say that that the use of that particular pharmaceutical in very young children is really unknown. It's a big question mark.
Okay, thank you.
Hi. I wanted to ask about amino acids. I don't know, if you guys think there could have any impact. I just haven't read much about it. I just wondered what you think about amino acids taken orally to see what happens.
I couldn't answer. I'm a big fan of amino acids, but
I've been sent some websites that report that they can use amino acids to cure children of neurological disorders, and I think including Angelman Syndrome. I've done some research on it. I can't find anything that supports that, and the websites, at least that were sent to me, had nothing on them that supported it. They just said, we've had success. these are, I would put these in the level of snake oil salesman. I would not waste your money. Most of the amino acids that your children need they're going to get through their diet. If they're missing an amino acid that's really important, then that usually causes a disorder that would have been picked up already in a metabolic situation. So I don't think supplementing amino acids is actually going to do anything majorly beneficial. But that's just my opinion.
No, that's fine. I just wanted to know. Thanks.
Hi again. I'm considering signing my son up for the levodopa trial, and I'm just wondering if that's really the best thing, if there is going to be a phase 3. I mean, I guess he's 4 now, so he might qualify, but not just individually for my son and my family, but globally for the whole community. What are the best choices for the newly diagnosed parents who have kids in this age range? Can you give us any sort of guidance on that? And then also, is there any way to make it easier? There's so much paperwork. We're so busy. We're so overwhelmed. I'm single. I tried to sign up actually through Baylor and played phone tag with the lady who's supposed to sign people up. And it dropped off the planet and I didn't get signed up. So I'm just, help.
Yeah, as far as the paperwork goes, I mean, you have to understand and this was a big eye-opener for me because I'm not a clinician. I'm a research scientist. So when the amount of paperwork that's involved on both ends to get somebody enrolled in a clinical trial is, a lot of that is federally regulated. You're not going to be able to get around it. The process is difficult because they want to make absolutely sure that this is, that you know what's going on, we know what's going on, and that there's not going to be any harm done, which is really what all of that is there for. As far as the levodopa clinical trial, there will be more clinical trials. I mean, you're going to have to determine in 5 years, if there's 4 clinical trials that are ongoing, you're going to have to determine which ones, if any, that you want to be involved in. I've said this before, that it's likely an effective therapy is going to involve multiple different types of therapeutics. It's important that we determine and understand through the clinical trials what role those particular therapeutics may have. So, I was talking about this earlier. You look around the room. There's a bell curve in height and hair color and everything that makes us unique. And we do this work in a mouse model that's all genetically identical. And so when you're looking at therapeutics in a population, you may have that particular therapeutic that works really good in this subset and doesn't work very good over here. So understanding how things like levodopa or minocycline or any other therapeutic that comes down the road, how that works in that particular individual is incredibly important. And the only way that you can do this is if you look at this therapeutic in a lot of people so that you can determine through to statistical analysis what groups of people that may work the best in. So, I'm not going to come up here and say, yes, you should do this, or no, you should do that, but I can say that it is important for us to look at all those different therapeutics, levodopa as well, to make that determination and see how that's going to be used as an effective overall treatment for the future. Is that, okay.
Yeah, that's great. What role does the RDCRN play in the clinical trials?
So the RDCRN is a group that has federal funding to promote and help clinical trials for rare disorders. So they have sites all over the country, which makes it a lot easier for parents to be able to take their children if there's multiple sites. They're coordinated so that they can do similar clinical trial in all of those different sites. And they know a lot about rare disorders. So they're not just working on Angelman syndrome per se, but Angelman syndrome is one of those rare disorders that they're looking at. So that entity and that network is incredibly important, especially when you're thinking about small clinical trials that are going to pop up in Angelman syndrome and that need to be expanded. they're there to do that, and they have the infrastructure, the expertise, and quite frankly, the ability to get a hold of federal money in order to conduct those incredibly expensive clinical trials. So to have them say that, yes, if the minocycline shows positive results at the end of your clinical trial, we'd be interested in expanding it, for us is huge. It's a huge relief, and it's a really important aspect for all the parents out there that have AS kids.
If I could just add, if the natural history study, the RDCRN people are trying desperately to get enough enrollees for the participants for the levodopa study. So if you've had any problem, if you're playing telephone tag, you could email Dr. Winhan Tan. They have a Facebook website or a Facebook site for the RDCRN. Just search on Angelman Natural History Study, and they will be glad to talk to you more about that. And would it also be fair to say, Dr. Weber, that Dr. Tan had volunteered to start drafting the protocol, and he's also leading the... Okay. Okay, so he heads up the levodopa study in the Natural History Consortium for Angelman Syndrome, and he is very supportive of the phase 2 trials for Dr. Weber's minocycline. So there's, some potential eventually synergy there as well. So it's a community working together, and I think that's the good news story.
So for those of you who are online or in the room and don't know who that was Sharon Claridge. She's one of our board members, and she's a science officer in the organization, and she's also our liaison with both the National Institutes of Health and the RDCRN. So she's done some really amazing work making sure that we can do things like expand the clinical trials, and she's done an amazing job for us. So, a question that we have online that is interesting. So, one of the things that's kind of clear is that our kids they have some similarities to them, and parents can say, I can diagnose a kid from, 100 yards because they all have similar facial issues or facial structures. And so this person's asking, does UBE3A affect that? Does it affect how they're presenting, or is this just all coincidence? This is sort of in a physical way. It's like your qualifying exam all over again.
I'll take a step. Yeah.
Yeah. See, I got really, Ann was supposed to be here.
I'm not aware, you guys interrupt, but I'm not aware of any studies that have been done that have looked at that. I don't know if there's anything in the literature. I know it's been described. If you look at, say, Down syndrome and other disorders, there are common features among them. So I suspect that UBE3A plays some role that is not understood in certain aspects of facial development and development of the mouth, and that's what you're picking up. That's my best guess.
I can say one of the most and often asked questions about that I get from parents is, can you look at the mice? Do they look like Angelman kids? And I'm here to tell you, the mice do not have any kind of facial feature that can distinguish them. So, which is good for us, because a lot of times if we're treating them with a drug, we want to blind the grad student, not literally blind them, but blind them to what the genotype is or what the drug interaction is, so that they don't really know. They go through all the tests, and then we can look later to see if there's significance. So, yeah, as far as I know, I don't know of any research, and we're certainly not looking at it in my laboratory.
Hi, I'm Michelle Cicerelli. I have a question in regards to the brain waves that they've seen. Is that better? My question is in regards to the brain waves and what they can see when they do an EEG on our children. One reason I'm asking is when my daughter was diagnosed with Angelman syndrome, the doctor said it was a characteristic of Angelman's for her to have these type of waves even when she's not having seizures, and that's what is causing her seizures, is the waves. So my question is that something that can help with also like diagnosing maybe where the problem is or what type of problem, and, like in regards to it from those waves.
This is exactly why we have Ann Anderson in with this group. And it's unfortunate she's not here because she's the perfect person to be able to answer that question. And she is an expert in EEG. She is really an expert in Angelman Syndrome in that aspect. And I think she has, and I can't really speak for her as far as, you know, what she's thinking, but one of the things that she wants to understand better is how the alterations in the EEG and that slow theta wave abnormality that they see in the EEG is related to the seizure phenotype. And I think through pharmacological intervention and some of the things that we want to do with the mouse model and eventually the rat model is to better understand that and to do that in the rat to kind of understand what those relationships are. So yeah, this is an area where we don't know about that and we don't really understand that, but it's an important part of understanding and developing any future therapeutic, and this is a reason why Anne is part of the group, and unfortunately she's not here to answer that question. See, I know what's going to happen. We're going to end this thing, and then you're all going to mob us up here and ask us a whole bunch of questions. Are there any online questions, Becky?
So I have a question for Dr. Siegel. How are you testing your zinc finger proteins? Are you doing them on cells or are you doing them in the mice?
Currently, we're testing them in mice. The cell culture models that we were using before actually were less informative than when we put it into the mice. And ultimately, the mouse is a more informative place to test them. So that's what we're trying to do now. And then through the FIRE grant, we're also going to try to establish neuronal cell cultures from human cells. So we'll be able to test it in a mouse model, soon in a rat model, but also in a human model, at least in a culture dish, not a brain.
Hello, I have an unrelated, not scientific question. Do you have any kind of like brochures or literature that we could pass on to possible donors?
So we've been writing this up and going through it. We will absolutely have information that people can pass on to donors. So what I would suggest that you do is write to myself or write to Paul or write to info@cureangelman.org. Tell us what you need and we'll make sure that you get it, because we have things written up in a way that we think will present it well for someone who maybe doesn't even think about science that much. But we can make it as easy or as complicated as you feel like the donor wants to see it.
Okay, thank you.
Mm-hmm.
And aside from money, what else can the community be looking for? You probably need computers. Maybe you need mice. Maybe somebody knows a mouse breeder. Start brainstorming and just keep letting us know what we can all do to make this fast, and we can help you with your fire. Bring more wood to your fire.
So we have another question online that comes up occasionally. So how do you guys feel like we're on the edge of finding a cure? Do you think it's going to be 5, 10, 20, 50? I know it's an unfair question, but that's the question.
So yeah, we've bounced this word cure around a lot, and I think it's a healthy discussion that we continue to have. I don't think that any of us would be doing what we're doing if we didn't think it was possible. It's hard to put a timeline on that. It's hard to understand how technology and scientific discovery are going to happen because we just don't know. I mean, we're doing as much as we can to make it as quickly as possible and to shorten up that time. Scott talked about the Human Genome Project costing $2 billion. And I remember when I was in grad school, they said it was going to get finished around 2050, I think is what they said. So, yeah, I'm older than you. So you know, with that kind of technology, it opens up not only opportunities but tools and availability for things that, you know, there's no way that we're going to know in the future. Yeah, to put a timeline on that, it would just be incredibly impossible and exceedingly difficult for us to do. However, I don't think that the difference that it has been made working with F.A.S.T and gearing your laboratory to do true translational research, you always have that in the back of your mind. How can we design the next best experiment to come up with a therapeutic that could lead to a cure? So those are all the things that you know, are always playing in the back of your mind when you're developing experiments, when you're thinking about new models, when you're thinking about fresh techniques and new ways to do this. So I don't think there's a specific timeline that we can set on this, but just say that, you know, whatever the time will take, we'll continue to work on it. I would love to cure this and go on to Alzheimer's disease, to tell you the truth. So, but we'll have to wait and see.
No, I don't.
So we have a comment from, I'm going to call her out. We're going to have a comment from Sarah Rorty online saying that she would like to remind you guys that AS will be 50 years old in 2015, so we would like that to be your target.
2015? We'll give it a shot.
Okay, let's get to work, everybody.
More questions? Okay, so I know you guys are going to probably come up here and mob them afterwards anyway, but I just wanted to say thank you to everybody. I know it's a long time to be sitting. It was fantastic having both the educational seminar yesterday and this today. I think we've talked about multiple ways to try to make lives better for our kids and for ourselves and our loved ones. And we hope you've all gotten something out of this and that you're walking away feeling like you have more information than you did when you walked in. That was our goal. And, you know, anything we can do to support you, you let us know. And find us on Facebook, go to the listserv, make sure that you're in the community and you're getting all the support you can get. And again, to help these lovely scientists, we're asking you to help us identify and raise funds to support this initiative so we can get more people into the initiative and we can get more hands in Angelman Syndrome research. I've said this to Paula a million times. I would like nothing better than for you guys to put me out of business. And when I say that, she always says, that's fine, then we'll find another one and we'll fix that. And but I'm serious, put me out of business. That being said, I would really like to thank the researchers today for answering all our questions and for being part of this. And thanks to everybody who was watching online and providing questions and keeping me very entertained while we were listening to the questions in the room. We were glad we can make this available. And let me remind you that we've taped everything. Ainsley's applauding wildly at the moment. We've taped both this session. We also taped the educational sessions. The educational session ran long, and Aaron Sheldon was kind enough to come down here and record a session with no one in the room so that we would get more of the literacy and education that we didn't get yesterday. These are going to be edited, and they're all going to be available to you online for free. So as soon as we have them available, we'll let you know. And I would also like to, hopefully you'll help me in thanking both the lovely crew who's been back here listening to all this and wondering why on earth they're learning about Angelman Syndrome. And Brian Thompson and Yahoo who made that possible for us. We have a comment from the audience who would like to make another final statement, I think.
This is on behalf of everyone. We wish you all the best of success.
And we'll take that. Thank you. All right. Everybody, thank you for coming. Safe travels on the way home, and hopefully we'll see you next year.